U.S. soybean farmers are proud, resilient. Some even call us, yep, we admit it, stubborn. We keep our promises to our families, our community, and to the earth. Sustainability isn't just a label, it's our livelihood. Our grandfathers remember when drought and over-farming in the 1930s turned our land to dust. That was about 75 years ago when our country put into place some of the world's most stringent resource protection laws. Today, U.S. soybean farmers are partners in trust to protect the land. We approach our mission as if the world depends on us, because in many ways, it does. More soybeans are grown here than anywhere in the world. Roughly 80 million metric tons a year are raised on over 30 million hectares of land. As a result of sound science and continued research over the past 30 years, we have increased soybean yields by 55% on roughly the same amount of land. U.S. soybean farmers have reduced carbon emissions, energy use, greenhouse gas, and soil erosion. We're licensed to use pesticides, insured to protect workers' rights and safety, and our assurance programs are verified. We're meeting U.S. laws and international sustainability standards. And to validate our compliance, a team of 3,000 career conservation agents throughout the country and continual verification through surveys, interviews, field observations, and electronic data that we're operating our farms in compliance with stringent environmental laws. Our commitment is more than just a neighborly agreement. Independent peer-reviewed research verifies U.S. soy is sustainably produced, with comprehensive data demonstrating that U.S. producers are compliant with the vast majority of our hectares already subject to annual audit by the USDA. And because we are driven by a commitment to continuous improvement, we're incorporating soil testing, plant tissue analysis, satellite positioning that precisely adjusts crop inputs, and much more. Why? To ensure that every link in soybean production is sustainable and to live up to our commitment as stewards of the land. If a U.S. soybean farm could tell its own story, it would be a legend, complete with dedication, resilience, compassion, and above all, successful, sustainable perseverance that lives to tell the story all over again. United States soy, verified sustainable. Learn more about our U.S. Soy Sustainability Pledge at ussec.org.
In the United States, there are about 2.2 million farms. We stand together and ready to continue to grow and produce the food that fuels the world.
Very good morning to you all. Welcome to the second day of the 73rd Scientific Sessions, Sri Lanka Veterinary Association. Very good morning to you all. Welcome to the second day of the 73rd scientific sections. We are conducting two parallel sessions at the moment, the clinical and the animal production. You are now logged in to the animal production session. I would like to hand over the proceedings to Dr. Chanaka Rabil, the session chair, and the panel of judges, Professor Alexander, Dr. Hemali Kotalawala, and Dr. Silva.
Thank you very much, Dr. Yushika. I take this opportunity to thank the organizing committee of the Sri Lanka Veterinary Association for inviting me to take proceedings as the chairperson of the animal production session. So as you know, there are 5 sessions, and this is the animal production session of the 73rd annual scientific sessions of the Sri Lanka Veterinary Association. So we will have 7 presentations, given the COVID situation. We have obtained pre-recorded recordings of all of them. However, there is one presenter here today. So we will have 6 recordings played and 1 live presentation. And I also want to thank the judges who have accepted the invitation in spite of their busy schedules. Dr. Hemali, DAPH, Professor Basil Alexander, and Dr. Susil Silva. So without further ado, so let me also, typically we have a recording, so we shouldn't run into time problems. We will dedicate 5 minutes for each Q&A session after the end of the presentation. And Vindhya, the only live presentation, please make sure your presentation will be limited to 10 minutes. Thank you very much. So with that, let us begin the first presentation, which is titled Effects of Fermented Juice of Epiphytic Lactic on Fermentation Quality of Maize Silage. The authors are MWDC Veeratunga, UWGDN Udagama, MPB Kumara Mahipala, and WMPB Veerasinghe. Okay, technical team, please carry on with the
Ladies
recording.
and gentlemen, I'm here to present the effects of fermentation juice of epiphytic lactic acid bacteria, FJLB, on fermentation quality of maize silage. Silage is a wet form of preservation of forage, which is mainly the microorganism utilize the available soluble carbohydrate under anaerobic condition, and they ferment lactic acid, which will reduce the pH of the forages and preserve for a period of time. But the tropical grasses are lack of soluble carbohydrate. Therefore, to enhance the fermentation quality, different additives were introduced in the past, especially the molasses. And after that, enzymes, acids, and lactic acid bacteria or the inoculants. The epiphytic lactic acid bacteria is one of them. That means the epiphytic lactic acid bacteria are known to be the naturally occurring bacteria on forages. So they have been used as an additive to enhance the fermentation quality of silage. So we are used that for to enhance the fermentation quality and evaluate sensory and fermentation quality parameters of maize silage ensiled with FJLB. The method was initially we designed the experiment and prepared the FJLB. After that, prepared the silage. Sampling was done and evaluate the sensory and fermentation qualities. So the experimental design consists with controls and the treatments, and each has 3 ratios, 0.5, 1, 1.5, and there were 3 replicates from each experiment, and altogether 18 silos. The FJLB is prepared using the guinea grass, where we harvest before flowering guinea grass and cut the leaves into small pieces and used 165 grams of grass and double the amount, that means 335 ml of distilled water, macerate it and filter it. The filtrate was taken and then add 3%, you can either 2%, we add here 3% of sugar to initiate the fermentation of the FGLB. And then we mix it well and pour it to the containers and keep them airtight and under the room temperature. So once we prepared, it was like dark green, and then the time goes, it appears to be brown color, dark brown. So we have to keep at least minimum 3 days to get the pH reduction and get the optimum fermentation characters. That means there as an additive of this FGLB to be used as an additive. So in our study, the pH was 3.85. The silage was prepared using the fodder maize at 90 days harvest. So we wilted overnight to reduce the moisture and then chopped into 1 to 1.5 inches. Then 1 kilogram of chopped maize was taken to prepare 1 silage. We designed ratios. We measure, you can see here. And at the same time, for the controls, we used distilled water and then mix and put it in the polythene bag, remove the air and tightly sealed for 45 days in room temperature. The sampling was done after 45 days. We open up the silage and mix well. Then subsample of 500 grams was taken for dry matter analysis and grind them and kept it in the containers until nutritive value analysis. The sensory parameters and the fermentation quality analysis was conducted. So sensory parameter was analyzed according to the field assessment guidelines once we open up the silage. The fermentation was evaluated by taking 10 grams of sample and measure it with distilled water around 70 ml. And pour it to the airtight containers and kept it under the refrigerator, kept it in the refrigerator under 4 centigrade. After that, filter the content. Then the filtrate is used to measure pH, lactic acid concentration, water-soluble carbohydrate and ammonia nitrogen level in our experiment. So the results were analyzed using one-way ANOVA with Minitab 16. The sensory parameters were evaluated as I told according to the field assessment guidelines. So basically we evaluate texture, color, odor, and the mold. So the color was similar in both controls and FGLB. Leaf is soft texture because it's maize, and the color is a little bit enhanced. Then controls dark olive green. The odor is again sweet, fruity, alcoholic aroma. So that indicates, according to the guidelines, the lactic acid fermentation, when it enhanced the fermentation, the good quality silage show those characters. So in our experiment, FGLB added silage shows that characters. So it revealed that FGLB has enhanced the fermentation quality of the silage. The dry matter content of the FGLB's added silages were decreased compared to the controls, and when you increase the FGLB concentration, dry matter concentration decreased. So that emphasized that there are heterofermentative and homofermentative bacteria. Heterofermentative bacteria produce other than lactic acid. So that may be the case. Sometimes carbon dioxide, hydrogen, and other gases they produce. So that will lead to decrease the dry matter content. And at the same time, water-soluble carbohydrate also decreased in the FGL-beared silages, which revealed that the utilization of water-soluble carbohydrate by the lactic acid bacteria in our experiment. So in that case, the increasing FJLB concentration has shown the increased lactic acid concentration in the experiment. So the highest observed in 1.5% compared to the controls. At the same time, when lactic acid concentration increased, pH reduction occurred. So the lowest pH observed in the similar 1.5%. And ammonia nitrogen is basically used to evaluate the degradation of protein. So actually it was negligible. It was less than 0.1 in both controls and treatments. That means the less spoilage of all the silages, as well as the good preservation of them. Therefore, we can say that application of FJLB in high concentration, that means 1.5 in our study, is improve the fermentation quality of maize silage. And also maize consists with high water-soluble carbohydrate content, and it is better we can apply in the tropical fodder silages to enhance the fermentation quality of silage. And in future, we will evaluate the nutritional quality of silage and also identify the potential bacterial species and then used as the industrial scale. I would like to thank all my supporters and thank you.
For a very interesting presentation indeed, which has a practical application in Sri Lanka. Importantly, with that, I open your presentation for discussion. Audience, do we have any questions? Sorry, online audience. Yeah, tablet online audience. There is one question from the live audience, Dr. Susanta Malavarache.
Thank you. Thank you, Doctor, for your interesting presentation. Can you hear me?
Yeah.
The question is, when you consider the commercial scale production, what are the parameters you need to look into that? Because keeping quality and the scale is very important when you go to the commercial scale. So do you have any idea about it?
Pardon, I didn't hear the first part.
Actually,
Can you repeat it first?
my question is, when you go to commercial scale production, so what are the areas you need to improve in that? Because keeping quality of the product and liability of the bacteria is very important. So have you done any test on that, or do you need to continue further?
Yeah, we need to continue further because the large scale they used prepared only this. So large scale usually they use that. So we initiate this process and we plan to identify the bacterial species, and then future we can utilize, we can give these back, after that as a powder or something, we can distribute to the farmers as well as to the industrial scale. That's what we are planning to do.
Okay. Thank you, Dr. Veeratunga. With that, may I ask the panel of judges if there are any questions? Yeah, please go ahead.
Thank you very much, Dr. Veeratunga. Very important area of research. It's a wonderful practical application. Can I ask us one single question? You talk about using lactobacilli. Do we have the lactobacilli concentration or the colony forming count in your setting? Did you measure
So it's difficult to hear.
Oh, sorry.
It's not clear actually.
Yeah. Sorry, did, did you measure the amount of lactobacilli in your product that you use, in your solution?
No, sir. Actually, I didn't have that capacity. We just measure the pH actually. So we are planning to isolate the organism. That means in different concentration and do further.
Because I think what matters is the amount of lactobacilli
Yeah.
in your, yeah. And,
Actually, we didn't have that facility at the moment.
and you said you use one-way ANOVA
Yeah.
for certain parameters, yes, but there are some non-parametric, no? The parameters
Yeah.
which are not distributed as well. So,
Yeah, I used only for the fermentative characters, not the other ones.
thank you very much.
Thank you, Dr. Susil. Do we have any further questions from the panel of judges? No? Okay. Thank you very much, Dr. Veeratunga. Since we do not have any further questions, we can conclude your presentation and move on to the second presentation, which is titled an estrus synchronization protocol using 17-alpha hydroxyprogesterone caprate on the pregnancy rate of goats, authored by S. Premaratna, T. De Silva, L. Fonseca, R. Sampath, K. M. Vimalasurya, E. Vikramasinghe, K. Premaratna, A. Olagadara, C. Gajavira, S. Disnaka, and I. Patirana. I believe the presenting author is I. Patirana. We have a recording of that. Please go ahead and play the recording.
Good morning, everyone. The title of the abstract is an estrus synchronization protocol using 17-alpha hydroxyprogesterone caprate pollutant on the pregnancy rate of goats. The plant breeding programs are really important in case of large-scale small ruminant farms in order to have proper management practices and proper farm planning. In Sri Lankan context, the local farmers need to go for expensive imported progesterone impregnated devices such as SIDA and sponges to synchronize their goats. However, these devices are not actually affordable for rural farmers in the country. Therefore, we thought of introducing a relatively low-cost diprogestogen as a solution to overcome this issue. The pollutant is used to prevent premature labor in pregnant women and exhibit a dipoaction. Actually, the pharmacokinetics of pollutant in goats was examined in India in a PhD thesis, as mentioned in the citation. So based on this research work, we conducted and we designed our experimental plan. The preliminary experiment was conducted at Aquaculture LDD farm. Initially, 32 crossbred dogs, which were separated into 4 groups randomly and kept for 2 weeks for flushing. The group 1 and 2 are given PGF2 alpha analog, the chlorprostanol, 120 micrograms per animal, 10 days apart. After the second dose of PGF2 alpha injection, 48 hours later, Group 1 animals were inseminated artificially, and Group 2 were introduced with buck. And Group 3 and 4, on day 0, pollutant 25 milligrams, which was dissolved in AD3E injectable solution, was given on day 0, and chlorprostanol 125 micrograms, as same as previous one, given at day 10. 48 hours later, after the PGF2 injection, Group 3 animals were inseminated artificially, and Group 4 animals were kept with buck. As the outcome of this experiment, we noted that the evidence of having a cervical opening in Group 3 animals compared to high compared to other group, group that is in Group 1. Also, the Group 4 animals showed 2 pregnancies after this synchronization program, which were kept with buck after prolutin and PGF2 injections. Based on the results, we went for experiment number 2 at Aquaculture same farm. In that experiment, we further modified that protocol, and 30 crossbred does were kept with high plane of nutrition, and on day 0 and day 10, respectively, we gave pollutant and PGF2 same dosage, and on the same day of PGF2 injection, on day 10, we introduced buck and kept for 1 week. And in this experiment, the results were quite improved than previous one. The total 18 does were pregnant out of this 30, and we obtained 4 twin birds. Based on the results of experiment number 2 at Aquaculture, we further improved that protocol with high plane of nutrition, and to investigate the efficacy of this protocol. There we used Sanan dose. Comparatively, in Aquaculture, this research was carried out in Alameda Farm, which is a dairy goat farm in intensively managed dairy goat farm. Compared to Aquaculture, the environment is quite good, and the animals were kept in high plane of nutrition, providing commercial cattle feed 700 grams per animal per day. Usually they gave 500 grams per animal per day, and mineral and vitamin premix aminol 15 grams per day per animal, and cod liver oil, that is 10 milliliters animal intramuscular orally for 2 weeks apart, and single shot of multivitamin injection 5 milliliters per animal per single shot, and vitamin E and selenium injection 2 milliliters per animal intramuscularly. Moving on to synchronization protocol, on day 0, 25 milligrams per animal were given pollutant, that is 0.1 milliliter of pollutant dissolved in 0.9 milliliter of oil-based vitamin AD3 injectable, and we injected intramuscularly. On day 10, PGF2 alpha analog, chlorprostanol, same dosage at Aquaculture, 125 micrograms per animal, given intramuscularly, and those were kept with Sanan bucks for 7 days for mating. Further, we obtained blood samples to measure serum progesterone concentrations on day 0, day 10, day 13, and day 31. And the progesterone levels were measured using an enzyme immunoassay. The pregnancy confirmation was done by ultrasound scanning on day 60. In the results, the progesterone assay, we got day 0, the depot action of the pollutant, and suddenly on day 13, the significant reduction of progesterone to be observed, and sharp increase of progesterone levels can be seen from day 13 to day 31. Almost all animals, that is 100% pregnancy rate, was obtained from this research. Out of, in this study, total 41 live births were recorded, and out of these 41, 12 twin births, that is 38.7%, and 19 single births were recorded. However, one doe, which was carrying full-term pregnancy, had died suddenly due to an intestinal torsion. Interestingly, based on farm records, the live twin pregnancies were significantly higher compared to the same month of the previous year. In this study period, total 14 twin births were obtained in the particular month. However, 11 out of these 14 were from the experiment. Last year, it was only one twin birth. In conclusion, so using this low-cost tested protocol, we can maximize the breeding efficiency of female goats in the field, actually, under high plane of nutrition. That will be further studied, and research are being carried out to develop a low-cost feeding regime to improve the nutritional plane and to perform artificial insemination, that is time AI, after the tested synchronization protocol. So we must thankful to Dr. Dhammika Pereira for his technical assistance during preliminary experiment which was conducted at Aquaculture farm. Thank you very much.
Okay, thank you very much, Dr. Samira Premaratne and the team for an excellent study. With that, I open your presentation for discussion. First of all, I don't have any online questions from the online audience yet. So I request if there are any questions from the current audience, and then we can move on to the judges. There doesn't look like there are any questions from the audience. So can I please ask the panel of judges if they have any questions?
Hello, Dr. Samir.
Yes, sir.
Sir. Yes, sir.
Yes. Yeah, that was a very good study, Dr. Samir. And I have a small question, like that you said the progesterone preparation, Prolutin, right? Prolutin.
Yes, exactly. Exactly correct.
Any experiments have been conducted using progesterone, like pharmacokinetics in goats, like, you know, we have commercial preparations already. They are already being used in the farms. Any the pharmacokinetic studies, like when you inject progesterone, how long it stays inside the goat?
Yes, sir. There is an literature survey. There was a literature survey we conducted. Actually, we have found out that there was a research in 2006, which was carried out by an Indian PhD student. They have studied the pharmacokinetics of goats, buffalo, and cattle in their study. And based on the results that they have obtained, we designed that protocol based on the levels of progesterone that will be remained in the blood in the goats.
So when you inject this drug, how long it will stay in the goat?
Sir, can I join? I'm Indranil.
Yes.
Yeah, the corresponding of the oath of the paper, can I join? Is it all right if I give you the information?
Yes,
Sure.
Yeah, the thing is, actually, as Dr. Samira mentioned, there is a very detailed kind of PhD thesis done by one Indian researcher that was done in 2006. And so they have used, I mean, even not only goats, even sheep, cow and buffalo as well. I mean, cattle and buffalo as well. So then it's a detailed study. And there they clearly mentioned, I mean, they have done frequent sampling and measured both progesterone and also this 7-hydroxyprogesterone. And based on pharmacokinetics, so it will go down. I mean, it will decline. It will be declined below 1 nanograms per milliliter after 10 days. So it will reach the peak around 4 or 5 days and will be waned off after 10 days. So that's why we thought to pick this, I mean, from 0 to day 10, and then to inject prostaglandin. So that is a very detailed study and available online. Yes.
Yes. But actually, progesterone has a negative. This is kind of, if you inject progesterone, it will have a negative effect. I mean, still it can, stay in the fat tissues, and when you give PG, still it has a negative effect on sperm motility and ovulation. Usually we don't inject progesterone in our experiments, so that's why I ask usually.
The only things are here, this is a kind of depot injection.
Orally, and you just remove it.
Yeah, this is a depot injection, and then the thing is that this is go below the normal concentration after 10 days. So then it will be, I mean, more or less equal to the normal body concentrations after 10 days. So based on the pharmacokinetics available.
Oh, okay, thank you. Okay, Dr. Susil has a question. Come on, go ahead, sir.
Yeah, thank you very much for the excellent presentation. I have a small question. I think when you have 100% pregnancy rate and X number of twin pregnancies, you might not need to have a control to compare with. But how appropriate for you to compare the results of this study with the previous years? I don't know, sir, your opinion.
Yeah, actually, sir, this is one of, I mean, this is a kind of series of studies. One of my PhD student is working on this under experimental conditions as well at here at maybe around, I mean, in Hambantota, the Valigatta Colombo University Center. And so this is a kind of series of studies, and we take appropriate control. But in this case, we tried control at Kottukachia based on the setup there, and then we found this protocol is successful, and then we thought of having many animals rather than getting a kind of negative control, separating those animals just to calculate the kidding rate and the, otherwise, we have to reduce the number of animals. So, based on the promising results from the preliminary experiments 1 and 2, we thought of sharing this information with the field vets. That is why we were targeting SLVA. Because, if the field vets try this and get kind of, if we can get kind of feedback from the field, it will be really grateful. And we are conducting, we will continue this under experimental conditions here in Hambantota.
Okay, yeah. Wonderful. Thank you very much, Dr. Indunil and the team. Since we have already run out of the allocated 5-minute time frame, we will move on to the next presentation. So the next presentation is titled Use of Vestibular Cytology as an Indicator for Estrus Detection and Artificial Insemination in Crossbred Goats, authored by MBD Lakmali, GDRK Pereira, RMC Deshapriya, BMS Munasinghe, and RMSBK Ranasinghe. I believe Dr. Ranasinghe will be presenting this, and we have the pre-recorded version. Please go ahead and play the recording.
Use of vulval cytology as an indicator for estrus detection and artificial insemination in crossbred goats. Introduction of my study, the improvement of reproductive efficiency will contribute to increase the production of meat and milk. The success rate of artificial insemination is low in Sri Lanka due to several reasons, such as poor detection of estrus and incorrect timing of AI. Vaginal cytology has been reported to be a sensitive indicator to determine the stages of estrus cycle in goat. This is my objective, to study vulval cytology as an indicator of estrus detection in crossbred goats. We'll go to the methodology. Study location, Udaperadeniya Veterinary Teaching Farm. Animals, 10 cycling crossbred does of same age, weight range between 40 to 50 kilogram body weight. All animals were fed grass and commercial feed. This is the estrus synchronization protocol of my study. On day 1, 20 milligram progesterone implants were inserted. Then day 20, all were removed, and same day 400 IU/PMSG was injected intramuscularly. Day 20, ultrasound scanning were done daily for 5 days. From day 1 to day 30, all animals' vulval impression smears were taken. This is the way I prepared smears. All animals were restrained in standing position. Vulva and perineum were cleaned. Vulval impression smears were taken by left hand. Vulval flaps open. The smears were taken from right hand. Staining. Smears were dried. Staining Ginza. 200 cells were counted in zigzag pattern. Statistic analysis. Different cell types were identified and percentages were calculated. The mean percentage of each cell type on each day was compared using repeated measures ANOVA in SPSS. Now see the results. Main cell types observed in smears. Actually, there were 6 types of cells, 4 types epithelials, and others are neutrophils and red blood cells. Superficial cells, keratinized cells, parabasal cells, and intermediate cells were the epithelial cells. Superficial cells, larger cells and polygonal in shape, nucleus elongated and dark. When considered keratinized cells, they were dead cells, anucleated with sharp edges. In the case of parabasal cells, they were rounded, nucleus rounded and large. Intermediate cells, polygonal in shape, borders are irregular, nuclei round. Finally, the neutrophils, subdivided into segmented neutrophils. Neutrophils contain a nucleus divided into 2 to 5 lobes. Average percentage of superficial cells From day 1 to day 30 of estrus cycle of crossbred goats, the maximum superficial cells was appeared on day 5. It was 82.8 plus or minus 4.5%. From day 1 to day 5, superficial cells were gradually increasing, then after gradually decreasing. No superficial cells were observed day 22-23. When considered keratinized cells, maximum percentage was in day 8. No keratinized cells were appeared from day 22 to 25. From day 1 to day 5, all percentages were below 20. Average percentage of parabasal cells in same duration from day 1 to day 9 gradually decline, slightly decline, but all percentages again below the 20%. Maximum in day 21. Finally, the intermediate cells percentages very clear from day 1 to day 6. Intermediate cells were not presented. Maximum in day 25. Here, images from ultrasound scanning from day 1, day of progesterone removal, to day 4. Actually, this scanning was done because to confirm the estrus and also to correlate the stage of estrus with the exfoliating cytology. Day 1, very tiny follicles could seen, and day 2, little bigger follicles, growing follicles, and maximum follicle size were noted in day 3. Day 4, there was no any follicles observed. Discussion of my study, vulval cytology observed during the estrus cycle of goats in this study is similar to the vaginal cytology described in other studies. If move to conclusion, during estrus of the studied crossbred goats, vulval cytology is dominated by superficial cells representing more than 70%. Intermediate cells are absent during estrus. Parabasal cells and keratinized cells are less than 20% in estrus. I would like to acknowledge Peradeniya University Research Grant number URG 2018-49B, as well as I would like to thank farm manager and staff of veterinary teaching farm Uda Peradeniya, and also like to thank academic and non-academic staff, Department of Basic Veterinary Sciences, Faculty of Veterinary Medicine and Animal Science. And thank you.
Thank you very much, Dr. Lakmali. So with that, I open your presentation for discussion. First of all, let's see if we have any questions from the audience. Yes, there is a question by Dr. Susantha Malavarachi.
Thank you for the presentation. I have a question that is, did you check the smear from the normal animal, non-estrus animal, and also the age of, what are the age group of that particular animal you tested?
It is after fourth, actually age between more than 2 and 5 years to 6 years. After third calving, Yes, after third KD.
Did you check the smear from normal and stress animal?
Actually, the selected all animals hold the cycle checked for 2 times, 2 cycles before the data collected the final time. So in that case, all the animals not without synchronized animals considered. The finally this time for this abstract contain data from all the synchronized one only.
Okay, thank you very much, Dr. Lakmali. Now I invite the panel of judges to raise any questions if they do have.
Dr. Lakmali.
Yes, sir.
Hello. Yeah, it's a good presentation. And I would like to ask a small question. Like in your slide, sometimes you say Easter cycle between 0 to 30 days of Easter cycle in goat. What do you mean by that? Usually about 21 days.
Yes, sir. So that means the collecting data for day 0 mean the day of insert the progesterone 20 milligram. That is not the Easter cycle actually. It is the day 0 mean the day of implant progesterone. Then the 30-day mean just after removing the implant devices. Then from there we take the samples up to 30 days.
Okay, yes. Yeah, 30 day of Easter cycle, you can't say like that. Yes.
Yes, sir. Yes.
Maybe 0 to 30 days, after your protocol.
Yes.
Dr. Hill has a question.
Thank you, Dr. Lakmali.
Yes, sir.
It's a very small question. This is like a differential cell count to decide the estrus, and when do you can do the insemination?
Yes.
Yeah.
Yes. Actually, if the superficial cells are prominent, that indicate the animal in pro-estrus just after coming to the estrus. Actually, in my study, same day I've taken the body temperature even. So after removing the progesterone, the second day, after 48 hours, body temperature was gone high. But it is sometimes more than 4 degrees. But I am not included into the abstract because I wanted to recheck it. So in the case of body temperature and the presence of superficial cells that indicate animal is come to heat. Then after the signs of increased temperature and appearance of superficial cells after 48 hours, we can do the AI.
What is the cutoff? It is 70%.
70% mean normally the gradually increase after day 3, day 4, gradually increasing and maximum in day 5. So after that decline. Normally in literature they're saying that the superficial cells more or less higher in the pro-estrus. So we have to ready for the AI just after that.
Or coupled with your temperature as well.
Yes, sir.
Okay, right. Thank you very much, Dr. Lakmali. So maybe, a small comment from me. So maybe in line with what Dr. Sushil's comments at the beginning, maybe your conclusion could have been, even on the abstract, this study shows that vaginal cytology can be used for this purpose, So then, it would be very well in line with the objective. Okay,
yes.
Thank you so much,
Sir,
Dr. Lakmali. So we have come to conclusion of your 5 minutes. Thank you very much. And with that, I will move on to the next presentation, which is titled Effect of Climate and Management Conditions on Production Parameters of Selected Commercial Layer Strains, authored by NMID Karunadasa and CMB Demetavava. Please go on with the presentation recording, please.
Hello, everyone. It's my privilege to do a scientific research on this 73rd annual scientific session in Sri Lanka Veterinary Association. I just want to tell you about my research. This is about effect of climate and management condition on production parameters of selected commercial layer strains. As this is the poultry-based research, I just want to tell you about some basic things. At present, the poultry industry is very developed, most developed livestock sector, and it's specialized with layer and broiler industries. And for the year 2019, the poultry sector contribute 0.38 GDP, which is 64% of the total contribution of Sri Lankan livestock. And here, the various commercial layer strains as well as the broiler strains have been imported to Sri Lanka from another countries, which can be used in different agroclimatic zones. And production performance of various commercial strains depend on the climatic condition and management conditions. Therefore, it is essential to compare the field level production performance of various commercial strains before recommending suitable strains for different management conditions. See, sometimes with these strains, they are giving their manual, and in there it's mentioned in their production level. But in field situation, we can't reach that production levels.
Okay. So I apologize for the technical failure on our side. We are going to restart the presentation from the beginning. The recording will be played from the beginning.
Hello everyone. It's my privilege to do a scientific research on this 73rd annual scientific session in Sri Lanka Veterinary Association. I just want to tell you about my research. This is about effect of climate and management condition on production parameters of selected commercial layer strains. As this is the poultry-based research, I just want to tell you about some basic things. At present, the poultry industry is very developed, most developed livestock sector, and it's specialized with layer and broiler industries. And for the year 2019, the poultry sector contribute 0.38 GDP, which is 64% of the total contribution of Sri Lankan livestock. And here, the various commercial layer strains as well as the broiler strains have been imported to Sri Lanka from another countries, which can be used in different agroclimatic zones. And production performance of various commercial strains depend on the climatic condition and management conditions. Therefore, it is essential to compare the field level production performance of various commercial strains before recommending suitable strains for different management conditions. See, sometimes with these strains, they are giving their manual, and in there it's mentioned in their production level. But in field situation, we can't reach that production levels with our management conditions as well as with the climate. So it is essential to look at the effect of these strains to recommending the strains into different climatic zones. And the basic objective of this study is to compare 3 commercial layer strains as Shaver Brown, Bowen Brown, and Lowman Brown on age at sexual maturity, in day production, and mortality percentage under intensive management in cold climate condition in Badulla district. Okay, other than that, to determine the effect of age of layers of different commercial layer strains on hen day production percentage and mortality percentage. Another objective of this study is to determine interaction effect of strain, age group, and location on hen day production and mortality percentage. And this is the place where I carried out my research. It's Bogananda Livestock Farm. It's located in Badulla district, close to Velimada area, and it is here. And for this area, the altitude of this area is 680 meters above mean sea level, and the annual rainfall is 1,400 millimeters. The average temperature is 21 Celsius. Average wind speed is 18 kilometers per hour. The total area of this farm is 88.8 hectares. These are the location of poultry houses. There are 5, and they are located quite very closely. This is the closer view, and these poultry houses are more or less same in structure. The roof height, the solid walls, the area of opening likewise, and the instruments used inside these poultry houses, those are more or less in each and every cages. And these are the main layer strains which reared during this period from 2014 to 2019. Those are Lowman Brown, Shaver Brown, and Bowen Brown. Other than this layer strain, I just want to tell you about the management practices which carried out in this farm. These poultry houses, they are open and they are managed in an intensively way. Some management conditions as the flow size, cage height, roofing material, spacing, feeding, those maintained in all poultry houses are same. The same management conditions are there in all poultry houses. And the vaccination programs are carried out to control infectious vessel disease and Newcastle disease for batches. And as biosecurity measure, mostly 1 or 2 laborers are involving the management practices in these poultry houses, such as collecting eggs, feeding, and watering. For this research, the data were collected to perform record database in Bogananda Farm. The records from 2014 to 2019 were collected, and those are calendar date, cage number, commercial strain, catch number, number of birds per cage per day, egg production on daily basis, and daily deaths during the production production period. And at this age of these layers are divided into basically the 6 groups depending on 150-day intervals, and below than 150 days it's 1, 150 days to 300 days it's 2. Likewise, this age group is divided into 6 groups basically, and this is how we analyze our data. For the age at sexual maturity was measured when the batch recorded 5% laying, and the hen day production percentage was calculated based on the 7,426 records, and total mortality during 150-day interval was used to calculate mortality percentage of the age group, and this is how we analyze those data. Before that, I just want to recall your memory on hen day production. Hen day production percentage is total number of eggs produced on the day divided by total number of hens present on the day multiplied by 100, and mortality percentage of the age group is total mortality during the 150-day interval divided by number of birds at start of 150-day interval multiplied by 100. And this is how we analyze our data. Analysis of variance procedure was carried out the ANOVA. And for the ANOVA, we take the strain of the poultry age group, cage location. Those are the fixed effect. We are not changing these effects, fixed. And dependent variables, hen day production, age at point of lay, mortality percentage. These are dependent variables. And for compare means, the Duncan's multiple range test was used. And this is our statistical model. Here, this one is hen day production, mu overall mean, the effect of strain, age, cage. These are interactions. Here, how you can see this the model. This is how you can see the results. The strain comparison on hen day production. This Lowen Brown day shows the highest hen day production percentage. It's 65.4, and Shea Brown's it's 58.6, and Bowen's Brown it's 50. This highest hen day production is from Lowen Brown strain. This is here strain comparison of point of lay. Based on the results, it doesn't have any significant difference among these strains on point of lay. Here the strain comparison of mortality. All 3 strains shows the more or less same mortality percentage. It doesn't have any significant difference. Here the age group versus hen day production. Here the age group second and third, they shows the highest hen day production percentage. Among that, the age group 3 shows the highest. But others, they doesn't have any significant difference. This is how we can show you the age group versus hen day production. The second age group is the highest hen day production. Age group versus mortality percentage, it doesn't show any significant difference in age groups and mortality percentage. As conclusion, I like to say the significant differences exist among layer strains with respect to production performance under cold conditions in Bornholm area. So the Lowen Brown strain shows the most superior performance among 3 strains, and hence we can recommend this Lowen Brown strain for the colder areas such as Valley Meadow. However, this overall performance of 3 strains are below their recommended performance standards, so This indicates that the special care in housing management is required to these cold regions to achieve the recommended performance standards of the commercial layers. These are the conclusions and recommendations. And that is the end. And thank you for your kind attention. And thank you very much. Have a nice day.
Thank you very much, Ms. Karuna Dasa, for a comprehensive study. With that, I open the presentation for discussion, and I want to remind the online audience, you are encouraged to ask questions by using the raise hand option, right? Then we can direct your questions appropriately. There are no online questions yet. So may I ask if the audience has any questions?
Hello.
From the judging panel.
Yeah. Hello.
Yes.
I have raised the hand. Cannot you see that?
I cannot see that, unfortunately.
That means this.
Is that Dr. Deshapriya?
Yeah.
Yeah, sir, go ahead, sir.
So thank you very much for the presentation. So your title says the management and the climatic condition related things. So I didn't see any climatic data or correlation between the production parameters and the climatic data. Can you explain this one?
Actually, sir, I just put about the temperature and other things. Actually, the previous topic is different, and after the reviewers comment, we just changed the topic. Here, we see the hen day production, the dependence of strain with hen day production.
So that means your study and the presentation title is different, or what you mean that?
We put another title actually, but with the reviewers comment, with less time, I have changed that title. But the basic thing is I check the hen day production performance with the strain basically. Depending on the strain, this hen day production also vary with that climate and management. So it's better to find a good strain to that area which suits with the production.
Okay, thank you very much, Professor Deshapriya. Do you have any other questions?
That's okay.
Okay, thank you, Professor Deshapriya. In that case, I can direct questions to the judging panel. We have a question from Dr. Hemali Kotalavar. Madam, please go ahead.
Actually, I also wanted to ask the same question as Professor Deshapriya. Yes, since you have not selected climate and management as your independent variables, and you have tested hen day production parameters as you have taken as the dependent variables. So you have not observed, your results say not no significant difference between the three strains. So what is your recommendation?
Sorry, madam. With climate, what is the recommendation regarding the strain?
Yes. Even within the same climatic condition. So
yes,
if it is, yes, you have not changed climate or management. So
yes.
That is not your independent variable, but your dependent variable was your production parameters, hen day production.
Yeah.
So you have tested age and strain against the production. Okay.
Yeah.
So
age and strain. Yes, ma'am.
Production. Yes. So yes, the title is different from what you have done here. And also, what is your recommendation based on your results?
Yes, with the strain, this production also little bit vary in different climatic zone. In this borderline area, it's quite the cold area. So among these three strains, the Lohmann Brown is the best strain that we can use as strain.
I think the question is, if there is no statistical significance, on what grounds do you say this strain is better than the other? Because there is no statistically significant difference. Is that right?
Yes. Yeah.
Yeah. So if it is not statistically significantly different, you cannot say A is better than B or vice versa.
We check the hen day production with strain, sir. With that, the highest egg day production shows by that strain.
Right. Yeah. Yes. Okay. So and even the iso mortality was for certain groups it was 3%. For other groups it was as high as 18%. So 6 times high. Yet there was no statistically significant difference. So I was wondering how that can be explained because 6 times difference. So that can theoretically happen if the sample number is small and all that. But at once it raised a question mark in my mind. So anyway, that's okay. So Dr. Sushil, is there a question?
Yes, for comment as well. Yeah, I think probably your title and the study probably doesn't matter. It doesn't matter. But we have probably analysis, what is the best suitable strain for Borderland farm. But we have tried, best suitable strain for Borderland farm, the way you manage Borderland, whoever. So apart from that, the egg production, egg size varies with age. It is very well documented up for different strains as well. And hen day production is, very low compared to the standards as well. So it's
Yes.
I hope you are not the manager of the Borderland farm.
No, sir. I was a veterinary surgeon in that area, the 2000 in the year 2018.
Don't share this publication with provincial director. All right, I was just joking. Right, thank you.
Okay, we have one more question from the audience. Yes, Dr. Santa, please.
Actually, I have a very unclear area in that your presentation, though it is a good try to understand the strain difference, because there are many character, what you call environmental factors and areas to be concerned when you are checking the performance in a poultry farm, because sometimes the mortality, the subclinical level of diseases and feed raw material variation, labor difference, and airflow, lot of things are to be considered. But here, the conclusion is not clear whether it is perfectly done according to the scientific factors. So, would you mind what you call conducting another trial to see whether the performance are based on the given parameters of the particular breeds or variant?
Yes, so here there are 5 poultry cages. They are located more closely, and for the management, mostly only the 2 workers are there. And feed also, we are giving the same way throughout this whole period. And that's why we put that. And I have those data, and later on I can analyze some other things as well.
Yes, better for that, and thank you for that.
Yes. Okay. Thank you very much, Dr. Karnada, sir, and everybody. So we will, since the time limit is passed, we will move on to the next presentation. So this is actually going to be presented in-house, as the presenter is here live. The title of the presentation is Effect of Egg Size and Flock Age on Post-Hatch Weight of Day-Old Chicks of Village Chicken, presented by UGVSS Kumara, CM Kirtiratna, MMDD Bandara, AB Amunugama, MBD Lakmali, and LGS Lukugalapati. And I invite Dr. UGVSS Kumara to present the findings live.
Good morning, everybody. We have 3 minutes more to good afternoon. Actually, this presentation is based on the research activities performed at Central Poultry Research Station, Karandagolla. But this study was to evaluate the effect of egg size and flock age on post-hatch weight of day-old chicks of village chicken in Sri Lanka. Indigenous chicken is much more popularized among backyard poultry farmers in Sri Lanka. Even though the village chicken has been reared mainly for the egg production, the surplus eggs and the old birds are used to provide meat as well in rural areas.
Linda, can you please pause? I think we're having a little bit of a technical difficulty. If you don't mind, because only 1 minute left, we'll have to restart from the beginning. Once the technical fault is fixed. ඒක නෙමෙයි මට නම් යන්නේ බ්ලැක් ස්ක්‍රීන් එකක් තියෙන්නේ. කළු පාට තියෙනවා. ප්‍රෝග්‍රෑම් තියෙනවා. Okay, I apologize for the technical lapse on our side. We are going to restart the presentation. Dr. India, please go ahead.
Thank you, sir. Good morning, everybody. Actually, this presentation is based on the research activities performed at Central Poultry Research Station. And this study was to evaluate the effect of egg size and flock age on post-hatch weight of day-old chicks of village chicken. Indigenous chicken rearing system is much popularized among backyard poultry farmers in Sri Lanka. Even though the village chicken is mainly reared for the egg purposes, the surplus cockerels and the old birds are used to provide meat as well in rural areas. Some backyard poultry farmers, they have their own mini hatcheries at the farm level. They are mainly catering the demand arises from the small scale farmers and low income and food deficient households. They reared mainly the chicken, village chicken, as a main protein rich source and income generator. Actually, as a result of that, the Central Poultry Research Station has been established to distribute good quality day-old chicks in order to distribute among backyard poultry farmers to improve their local chickens and the productivity. They are issuing the parent stocks for the hatchery owners, and they are issuing layers for the farmers to maintain their flock level at their farms level. Next one. Since the weight of day-old chicks is a very important factor to determine the egg production and the selling weight, it's more used to gain economical benefit from the poultry farming systems. According to the common opinion among poultry farmers, the large eggs produce the large chicks. These large chicks are stronger and grow more rapidly than the chicks resulted from the smaller eggs. There are some studies have shown that the hatch weight of chicks is not always positively correlated with the post-hatch performance of the chicks, because the presence of excess amount of yolk at the time of hatching, they shows the poor later performance of the resulted chicks. There are many studies have been found to evaluate the relationship between the egg size and flock age on day-old chick weight on commercial layers, the well-established commercial layers and the broilers. But in Sri Lanka, there is limited information on village chicken. Therefore, the objective of this study was to investigate the effect of egg size and the flock age on weight of day-old chicks of village chicken at Central Poultry Research Station. It's under the VRI and Department of Animal Production and Health. In methodology, we have identified 3 separate batches representing 3 age groups: 20 weeks of age, 32 weeks of age, and 45 weeks of age of village chicken. For the easy explanation, I have taken one batch as an example. From this batch, totally we had to collect 300 eggs. For that, 50 eggs were collected by weekly interval for 6 weeks period. For the eggs collection, eggs were collected on very first day of each week, and ultimately we had 300 eggs with us for each batch. At the time of collection, we excluded all the deformities of the eggs. It means the egg had to be clean and smooth thin surface, no cracks at all. All the deformities eggs were excluded. Then the eggs were categorized according to the egg weight. Here we have identified 3 different eggs categories, the weight categories: heavy eggs, average eggs, and light eggs. Here I have followed the Franco's method in 2010 for the categorization. Then after the egg collection, I wanted to study, is there any significant difference among these 3 categories? Therefore, and also I wanted to study, is there any significant difference among egg categories within the group and in between 3 batches. Therefore, the data was analyzed using the ANOVA at the 0.05 level of significance. Then after categorization, the eggs were transferred to the setter. The setter condition was 60% relative humidity and the 100% centigrade degree of Fahrenheit. Then after the setter, on 18 days of incubation, the eggs were candled to detect the fertile eggs. Then the cannulin detects bloody whites, blood spots, or meat spot and enables observation of the germ development. After that, the only fertile eggs were transferred to the hatcher. The hatcher condition was the 80% relative humidity and the 99 degree of Fahrenheit. In here, we have used pedigree boxes to keep the eggs within the hatcher machine for easy identification at the time of hatching. Then on 21st day on the incubation, after the incubation, the chicks were resulted. Then the weight of day-old chicks were measured and these resulted chicks were categorized according to the previous eggs group. It means the chicks resulted from the heavy eggs, the chicks resulted from the average eggs and from the light eggs. Then as previous, then I wanted to see is there any significant difference among these resulted chicks from the three different egg categories within the batch and in between the three batches. Therefore, the data were analyzed using the ANOVA at the level of 0.05 significance, and the differences of day-old chicks weight within and between batches were calculated. To find the relationship between the weight of day-old chicks and the egg weight, the correlation coefficients were determined. In here, okay. To check residual yolk effect, the moisture losses and the hatchability were determined. These are my results. According to the results, we can say there is a significant difference of mean egg weight in three batches, and there is a significant difference of mean day-old chick weight in 3 different batches. And also, no sir. Thank you. So the large eggs produce heavy chicks from batch number 3. It means the old flock. Then the small eggs produce small chicks from the batch number 1. It means the young flock. And also, according to the results, we can say there are significant differences among the egg categories: large eggs, medium eggs, and small eggs within the batch and in between the batches. And also, there are significant differences among resulted chicks weight within the batch and in between the batches. Yes. This graph also shows a strong positive correlation between the mean egg weight and the mean chick weight. According to the results, we can say the smaller chicks produced by the small eggs by young flock and the heavier chicks produced by large eggs from old flock. Therefore, according to the results, the egg weight influenced by breeder age of the village chickens and the weight of day-old chicks influenced by the egg weight. During the experimental period, we have observed that the ideal level of moisture losses and the hatchability percentage. Then we can say there were no residual yolk effect on resulted chicks as well. So at last we can, according to the results, we have found that the chick weight has been improved with the breeder age and egg weight with ideal level of moisture loss and the hatchability percentage. At last, the study concluded that the chick weight improved with the incubation of larger eggs. Therefore, the weight of day-old chick is a very important factor to gain early mature body weight. Therefore, we can use the larger size eggs should be selected for incubation to produce heavy chicks to get more economical benefit from the backyard poultry farmers as in the other well-established commercial breeds. But at last, I recommended to evaluate the effect of egg size on more production performance parameters of resulted chicks to get more economic benefit from the resulted chicks of village chicken, such as body weight gain, feed conversion ratio, feed intake, total egg production, etc. These are my references. Thank you. Thank you.
Okay, wonderful. Thank you, Dr. Vindya, for finishing your presentation on time. With that, I open the presentation for discussion. I don't see any questions online. Online audience, please use the raise hand option if you have any questions. And in the meantime, do we have any questions from the audience? No questions from the audience. Do we have any? We do have a question from the audience. Dr. Jiran Sadrasinghe, please go ahead.
Kanchana, thank you very much. And I'm also thankful for you conducting this research in the domestic birds and the village chicken. I think this is one of the first study conducted. And there are some concerns on this research for me, and I just want to give some suggestions. The first one is the fertility. When you check at 18 days, it is not actually the fertility, it is apparent fertility, because we have done only the canneling. Because so there can be early dates, right? Middle dates, but we don't see that. We have not done the breakout analysis. So that should be the apparent fertility. And normally it's a well-known fact that from the egg weight, the chick weight is around 66.66. And also in the breeders, right, when the age goes up, the egg size also goes up. And the large, older flocks produce large eggs, and the small young flocks produce the small eggs. And also the farmers, they like the bigger chicks because they think that they will grow fast. But on the other hand, the immunity side and all, when you check the disease, resistant and all, the young flocks give the healthy chicks because they have more antibody titers, maternal antibody titers coming from the parents, because the vaccination programs and everything, the titer levels in the younger flocks are higher than the older flocks. So in that case, younger flocks, they are more healthy and the mortality rates are lower in the field. And the second suggestion is when you check the hatch weight, right? So it's better to conduct the chick quality score also, because the moisture loss and everything, you can have the bigger chicks, but there is a score called Pascal score, right? You have to check the red hawks, and also you have to check the navel, open navel, and the string navel, and the quality of the chicks. So the quality of the chicks affects the direct parameters that you are using in the hatcher or the incubator, where the moisture loss and also the temperature. So the chicks, the hatchability will be very good, but still the chick quality can be compromised. So those two things you have to consider. So it's better if you can include that chick quality parameters in this study as well.
Thank you, sir, for your suggestions.
Thank you, Dr. Sadrasinghe. Do we have any other questions from the audience? Yeah, Dr. Sushil Silva has a question.
Yeah, village chickens, how close are they genetically?
In Sri Lanka, sir, we can't. Yes, I used the village chicken available in the Central Port Research Station, Karandagola. Actually, we can't say these animals are actually indigenous chickens, because at time to time they have performed some crossbreeding activities, but the plumage color and the phenotypically we can say that they are more or same to the indigenous chickens. But genetical analysis, I don't think, because I didn't find any articles in genetic analysis in village chicken Sri Lanka.
I know at Karandagola, you have cave crosses, you have naked,
Yes.
When you select, at least did you do some selection?
Yes, at the experimental time, I removed the naked neck chickens because they are phenotypically different among the other village chicken. I selected the birds who are phenotypically similar. Similar mean the plumage color are different in color, and actually phenotypically selection were there.
That's actually what I meant. Not serious genetic analysis. At least they are close enough. No? Yes.
Okay, thank you. One last minute. Any further questions? No? Okay, great. Thank you very much, Dr. Vindya. Now we move on to the penultimate presentation of the animal production session, titled Artificial Insemination of Sheep in Sri Lanka, a Successful Preliminary Study. authored by S. Premaratna, S. Kalugovila, C. Kannangara, S. Disnaka, GDRK Pereira, and MPS Magamage. I believe it is being presented by Dr. Samira Premaratna. Please play the recording whenever you're ready. Thank you.
Good morning, everyone. The title of my abstract is Artificial Insemination of Sheep in Sri Lanka, a Successful Preliminary Study. The sheep farming is not much popular in the country. However, there are around 11,500 sheep according to the statistics in Sri Lanka, and approximately 7,000 are reared in NLDB farms. That is representing more than 50% of the Sri Lankan sheep population. Red Madras, Bannur, and Jaffna local are the common breeds available in NLDB farms as well as other farms in the country. The sheep are mainly reared for meat, manure, blood harvesting, and issuing breeded materials in NLDB breeder farms. However, the slow growth of existing sheep population is mainly due to inbreeding, and there is no genetic upgrading programs were conducted up to now. In National Livestock Development Boards, mainly sheep are reared in Coconut Triangle and Dry Zone farms, namely Horokale, Kottukachchiya, Oyamadua, Rukkatana, and Andigama. In Coconut Triangle farms, sheep are reared under coconut and other livestock species as integrated farms. The major objective of the AI program is genetic upgrading of existing sheep population in National Livestock Development Board. There are two types of artificial insemination methods available for AI in sheep. The one is laparoscopic method, which is shown in the picture. Actually, this is not used in Sri Lanka and other countries. They do laparoscopic AIs for sheep. The other method is transcervical method. That is the method that we used in our AI program. In the Horokale farm, the total population is approximately 3,300. And in this program, we use initially healthy crossbred, that is Red Madras and Bannur, total 114 animals. And before doing estrus synchronization, we kept them under good plan of nutrition as a flushing up ration that we provided during the 2 weeks time with commercial cattle feed, 200 grams per animal per day, and vitamin and mineral premix, 15 grams per animal per day, and vitamin E and selenium injections, 3 ml per animal. Moving on to estrus synchronization and artificial insemination, on day 0, we applied or we inserted easy breed SIDA transvaginal application we did, and that SIDA comprised of 300 milligrams of progesterone, and On day 14, the SIDA was removed from the animals. Now, after 54 to 56 hours from the removal of SIDA, the particular ewes were inseminated using frozen thawed imported Australian white semen from Australia. And finally, we inseminated 98 ewes. Actually, the 16 SIDAs were dropped between day 0 to day 40. And the pregnancy diagnosis was conducted using ultrasound scanner after 45 to 60 days of insemination. This is how the SIDA is placed in the applicator prior to insertion. And in this picture, you can see how the already set up SIDA is applied to vagina. Some photographs of ultrasound scanning. And the total AI success rate was 21.4%. Actually, 19 live births we obtained. Out of 19, 11 were males and 8 were females. The 2 abortions were recorded. And one lamb died, actually, because of the emphysematous abomascitis at the age of 1 month. And the average gestation length was 156 days. And the average birth weight of lamb was 2.6 kilograms. Some lambs we got from this program. In this picture, you can see. However, this is the first record of conducting AI in sheep in the country as a genetic upgrading program. In this study, we obtained 13 lambs. Actually, currently they are at the breedable age, and 8 rams will be used to upgrade our existing sheep flock. And this program showed that the feasibility of conducting artificial insemination in sheep in local conditions and the potential of developing best fitted, economically viable sheep breeds to the country to support the growth of sheep population in Sri Lanka. I must thankful to Livestock Division of the Line Ministry and staff members of Forakali Farm of National Livestock Development Board for the dedication to conduct this AI program. Thank you.
Thank you very much, Dr. Premaratne, for a nice presentation and a well-designed study. I hereby open the presentation for discussion. Do we have any questions at the audience? Yes, we do. Dr. Susana Malavarati, please go ahead.
Yeah, actually, it is a very innovative presentation and finding. And I have 2 questions. That is, one thing is to know how did you measure the body condition score and the body weight of the each female before you that vitamin and mineral premix? Did you check the what do you call ingredient content to meet the body requirement, energy requirement before you formulate the feed?
Yeah.
Dr. Premaratne,
thank you.
Can you hear?
Yeah, I can hear, but the voice is not a little bit unclear, interrupting, I think.
Okay, I'll ask the question again. I just want to know two things. That one is that did you check the body condition score and the body weight of each female before AI? And the second one is when you say you use vitamin and mineral premixes to formulate the feed, did you check the ingredient content of each vitamin and mineral?
Yeah, actually, so we selected animals from our flock of sheep in Horakalle farm. So basically, we used the body condition is actually when we got the 1 to 5 scale. Actually, they are around medium, right? For which are eligible for breeding and other things, and so that is how we selected. And the 1 to 3 parities. The user belongs to 1 to 3rd parities. So then the second one is the cattle feed. We used commercial cattle feed, and the mineral and vitamin premix that we used, the one which we produce at the Kottukache farm. So we produce vitamin and mineral premix. So they are at Kottukache farm for NLDB animals, and that is formulated by Veterinary Research Institute, and we use that.
Okay, thank you very much,
Thank you.
Dr. Premaratna. Do we have any questions from the panel of judges? Yes, there is a question by Dr. Hemali Kotrala. Yes, madam, please go ahead.
Yes. Did you observe any difference between AI success rate and the natural service success rate?
Yeah,
the
madam,
farm?
yeah. So in our farm, so the natural success rate is quite, so we are rearing as flocks. So usually we reared 300 or 300 to 400 per animals per flock. Usually the pregnancy rate would be 70 to 80% when we do ultrasound scanning after they introduce the birth.
So yes, so significant difference is there. So
Yeah, exactly.
my next question is about the cost of how did you get semen?
Yeah, we the semen imported from Australia through Line Ministry.
So what is the cost per one dose? Do you have any?
Yeah, it is around nearly, as far as I remember, it is around 4,500 nearly per dose.
Okay, thank you.
Thank you. The continuation of that question, I presume that 70 to 80% versus 20, that is an expected difference. But Samir, how does this 21.4 compare to the published data available from other countries?
Yeah, actually for sheep, so the sheep are quite different. So there are some light sensitive, and in other countries they use for with the time and the climatic conditions and the daylight availability of the countries. Usually the pregnancy rates were quite different with laparoscopic artificial insemination and both transcervical methods. So it depends on the countries, I think. So hopefully to continue in Sri Lanka further, we'll see the results in future.
Right. So you don't have a ballpark figure of the other rates, huh? Is it 40%, 50%?
Yeah, ranging from 40 to 50%. That depends on the light availability, daylight content of the country, and the climatic conditions and all.
Okay, thank you very much, Samira. Do we have any questions from the online audience? I don't see a single question coming from the online audience. Raise hand. Right. So thank you very much. And then we move on to the last presentation of the animal production session, which is new dimensions of swine breeding through artificial insemination in Sri Lanka. authored again by S. Premaratna, S. Tisnaka, G.D.R.K. Pereira, and M.P.S. Magamage. Please play the recording whenever you are ready.
Good morning, everyone. The title of my abstract is New Dimensions of Swine Breeding Through Artificial Insemination in Sri Lanka. In Sri Lanka, mainly the pig population can be seen from Kalathura to Puttlam area, where the most pig farms and pigs are concentrated, other than other areas of the country, and mainly which is commonly known as pig belt. And in Sri Lanka, there are about 165,000 pigs are available according to the statistics, and there are breeder and fattener farms available in the country. Among breeder farms, there are nuclear farms, multiplier and commercial child farms. And the large white, landrace, and Duroc are the major pig breeds reared in Sri Lanka. In this slide also illustrates the distribution of animals around the country, and major numbers are located, and major population, the population is congested, especially as I earlier said, from Kalathura to Puttlam, the coastal belt. Let's see the suppressors of swine farming in the country. The major constraints are lack of quality breeding materials to the farmers, and also the extension services and technical personnel are limited for this industry, which leads to poor planning and lack of motivation of farmers, and which creates some environmental issues also, and also high feed cost already affect the pig industry in the country as other livestock industries. And there are some biosecurity issues and the religious influence also there in the country, which suppress the growth of swine breeding and fattening in Sri Lanka. In the Horokele farm, it maintains imported generation of pigs from Canada and USA, mainly 120 purebred sow operation is there. It is the nucleus herd of the country. It consists of 50 large white, 50 landrace, and 20 Duroc sows. And breeding is conducted both natural service and artificial insemination. And this is the breeding plan of the country. And in Horokele farm, we reared, as I earlier said, a large white landrace and Duroc. So we cross large white landrace and produce large white landrace crosses, and which we issue to the local farmers with pure Duroc male animals. So the ultimate target is to produce three-way crossbred fattener to local farm. The major objective of this AI program at Horokele using defrozen semen is preventing breeding and maintaining genetic diversity of the replacement herd, and supplying of quality breeding materials to local farmers. That will be the ultimate target. This AI program was conducted from 2018 to 2020, and heat detection was conducted using visual signs of heat stress and applying back pressure. And before conducting AI, it's important to know the preparation of semen extender, which is quite different from other animals. So there is an extender pack containing 48 grams, which will be dissolved in autoclave distilled water under 35 degrees centigrade. That pack dissolved in 1 liter of autoclave distilled water is sufficient to prepare 12 numbers of bottles. And finally, once prepared, those bottles should be stored under freezing conditions. Once prepared, they extend the bottles, and before going to AI, it should be thawed up to 20 degrees centigrade. And the frozen semen should be thawed in 50 degrees centigrade for 45 seconds in a water bath, and should be mixed with that extender bottle prior to inseminate. And until insemination, the dissolved semen solution should be kept at 20 degrees centigrade, and it should be inseminated as soon as possible. The timing of the AI, in sows, it should be 33 to 36 hours after detection of standing estrus, and gills, it was 29 to 32 hours after heat detection. Altogether, in this program, 116 AIs were conducted, and there was the tentative pregnancy diagnosis was carried out by looking at the non-returning to estrus after 18 to 24 days post artificial insemination, and physiological differences of inseminated animals. You can see the applied back pressure to realize whether the animal is in proper heat or not. So you can gently and you can push the backside as shown in the picture. The semen is stored under liquid nitrogen, minus 196 degrees centigrade, and how the extender solution is prepared. And this is once prepared, the extender solution is something like this in the bottle, and we can store this in the freezer. And we are conducting AI using this type of catheters, and the tip of the catheter is lubricated through using non-spermicidal gel, and vulva, the external genitalia, should be cleaned well using tissues. And finally, we inseminate. The AI success rate was 31% approximately, and we obtained total 36 numbers of furrowings, and out of this 17 were large white, 15 were landrace, and 4 were zero. The average litter size was around 7.8 and 7.8, and the 8.5 in large white, 7.6 in landrace, 6.2 in zero, and the male to female ratio among offspring was 54 to 46, and in large white, it was 61 to 39. Landrace, it was 46 to 54, and 44 to 56 in Europe. This AI program was shown that the feasibility of conducting AI in swine breeding farms under local conditions with minimal technical intervention, and it shows that the potential of preventing inbreeding and maintaining the hybrid vigor of the animals. Especially, I must thankful to Dr. Chindana Karunaratne for his technical assistance, and as well as Dr. Chamila Perani, staff of VIC Chilau, for the enormous support, and Division of Livestock Development in the Line Ministry, and staff members of Horakale Farm for their dedication to fulfill this program a success. Thank you.
Thank you very much, Dr. Premaratna. You must be a really tired man after having done 3 presentations. Good thing it was all recorded, pre-recorded. So
thank you very much.
With that, I'm opening the discussion up for Q&A session. Do we have any questions from the audience? We do have a question. Oh, from Dr. Susanta Mallavarachi this time. Thank you, sir, for asking questions and keeping it live. Yes, please carry on.
Actually, it is very interesting, and as mentioned that he may be tired, but really appreciating that good effort. And I know when you compare the success rate, it is a bit low, but the effort is very good. My question is now, when you apply the back pressure to identify the heat, how do you measure that the particular animal is in the correct timing? Or how do you judge whether that animal in the early stage or early hours or late hours? May I know the comment from you?
Yeah, exactly. Good. So once we wean the animals, especially sows, so we keep them in a particular pen, group pen. So we are looking for heat, because usually the sows are coming to heat within 7 days, actually in good conditions. So that sows will be frequently monitored for coming into stress, and they show some visual signs and everything. And when the animal is close to a boar, they get induced also for heat, and they show some behavioral changes and everything, because we feed them 2 times daily, and there are supervisors and they are for observing heat, and other workers. So then based on this finding, so we determine the exact timing. Actually, sometimes it can be missed, because that as example, I mean, so that is how we determine our time of standing heat, and then we apply back pressure and check whether the animal is for heat. In swine, actually, so the estrus to onset of ovulation, the time is quite have variation. Actually, it is in sows 10 hours to 58 hours. Once show the heat, then the ovulation occurs between 10 to 58 hours. And gills, it will be 16 to 56 hours later. So that is the thing behind that.
Okay, thank you, Dr. Premaratna. I hope the answer was clear, Dr. Malvarachi. Yes. Then I direct the question opportunity to the panel of judges. Do we have any questions for Dr. Premaratna?
Yeah.
Yes, there is a question from Dr. Susil.
Yeah, just a small comment. I think, thank you very much for making many presentations. Good to see young vets.
Yes, exactly.
Contributing hugely. And yes, for your previous presentation for sheep, I think it's about 20,000 per one pregnancy. And for this one, what is the cost? I know cost is very immaterial for your sows and for sheep as well, because the intention is to reduce inbreeding. I know you don't have a lot of stocks at Horakale. What is the cost roughly? Last one you said 4,000 for insemination and roughly around 20% success rate. That is mathematically it's about 20,000 per one pregnancy. What is per sows? Now 1/3 is the success rate? What is the
Yeah, actually, the cost is, as far as I remember, the cost is quite similar. In south for swine, the cost will be around 5,000 to 6,000 nearly, as far as I remember, because we imported a couple of years ago the semen stock. So that is the amount I remember.
yeah, it's well worth rather than importing animals? I think to keep the
so even if it is 20,000, let's say 10 piglets, it's 2,000 per piglet. I think that's,
and they don't have much larger stocks either. No, you have
Yeah.
about 30 house from US long time ago. I don't know whether you have introduced new blood after that.
Yeah, actually, if the cost is about, let's say, 10,000, so we get nearly around 1 lakh from each farrowing. So actually, we the cost is very low compared to that part. So one piglet costly around 11,000 actually. So as a government organization, we provide with low cost without getting any much profit to the farmers. So let's say that one animal is 10,000. So the cost is around one animal, the semen dose.
And on top of that, indirect profit of keeping the population,
avoiding inbreeding, things like that, genetic gain. Yes, yes. Okay, thank you very much, everybody. Thank you, Dr. Premaratna and all the presenters for a successful session, animal production session, which is now officially can be concluded. So we had a variety of presentations ranging from silage to, estrus synchronization in goats, to vaginal cytology in goats, to commercial layers, to village chicken, and then finally artificial insemination in sheep and swine. I wish there was more cattle oral presentations. Unfortunately, there are none other than the silage presentation. So just a comment, as a student, as a PhD student, one of the comments I got from my presentations was, I had too much information on my slide. Sometimes people ask me, if you are not going to discuss that thing, why are you having your slide? So I saw in certain slides, they were too busy, too much data. You only wanted to highlight, let's say, 3 pieces of data, but there's 20, right? So just a general comment. So don't make your slides too busy. Just have the ones you really want to emphasize, so that makes it clearer for the audience and the judges, right? So other than that, I see a dramatic improvement in the quality of presentations over the years, right? And again, I want to thank the authors, especially we see 3 or more presentations from the NLDB group, right? So keep up your research, keep sending these abstracts to the SLV, and I also want to emphasize, don't stop there. Try to publish these as full papers on SLVJ or beyond that even, right? And that those will be invaluable for the development of the livestock sector altogether. So with that, I conclude the session, and I want to thank the authors, the presenters, the online audience, as well as the handful audience we are having in-house here today. And I want to especially thank the panel of judges, in spite of their busy schedules, making time to attend the 73rd annual scientific sessions to make it successful. So judges, please hand me over your mark at the end of the session, and those will be used to evaluate the presentations and to select the winner of the the prize for the animal production session. Thank you very much. And with that, I hand over proceedings to Dr. Yushika.
Well, thank you all for such an interactive session. And this is the end of the animal production session for today. We'll be back with wildlife and aquaculture session with 12 more interesting presentations. Hoping to see you with the same enthusiasm at 1:15. good afternoon to you all. We are actually having a small delay, so we are starting the session at 1:30. Bear with us till then. Thank you.
U.S. soybean farmers are proud, resilient. Some even call us, yep, we admit it, stubborn. We keep our promises to our families, our community, and to the earth. Sustainability isn't just a label, it's our livelihood. Our grandfathers remember when drought and over-farming in the 1930s turned our land to dust. That was about 75 years ago when our country put into place some of the world's most stringent resource protection laws. Today, U.S. soybean farmers are partners in trust to protect the land. We approach our mission as if the world depends on us, because in many ways, it does. More soybeans are grown here than anywhere in the world. Roughly 80 million metric tons a year are raised on over 30 million hectares of land. As a result of sound science and continued research over the past 30 years, we have increased soybean yields by 55% on roughly the same amount of land. U.S. soybean farmers have reduced carbon emissions, energy use, greenhouse gas, and soil erosion. We're licensed to use pesticides, insured to protect workers' rights and safety, and our assurance programs are verified. We're meeting U.S. laws and international sustainability standards. And to validate our compliance, a team of 3,000 career conservation agents throughout the country and continual verification through surveys, interviews, field observations, and electronic data that we're operating our farms in compliance with stringent environmental laws. Our commitment is more than just a neighborly agreement. Independent peer-reviewed research verifies U.S. soy is sustainably produced, with comprehensive data demonstrating that U.S. producers are compliant with the vast majority of our hectares already subject to annual audit by the USDA. And because we are driven by a commitment to continuous improvement, we're incorporating soil testing, plant tissue analysis, satellite positioning that precisely adjusts crop inputs, and much more. Why? To ensure that every link in soybean production is sustainable and to live up to our commitment as stewards of the land. If a U.S. soybean farm could tell its own story, it would be a legend, complete with dedication, resilience, compassion, and above all, successful, sustainable perseverance that lives to tell the story all over again. United States Soy, verified sustainable. Learn more about our U.S. Soy Sustainability Pledge at ussec.org.
In the United States, there are about 2.2 million farms. We stand together and ready to continue to grow and produce the food that fuels the world.
U.S. soybean farmers are proud, resilient. Some even call us, yep, we admit it, stubborn. We keep our promises to our families, our community, and to the earth. Sustainability isn't just a label, it's our livelihood. Our grandfathers remember when drought and over-farming in the 1930s turned our land to dust. That was about 75 years ago when our country put into place some of the world's most stringent resource protection laws. Today, U.S. soybean farmers are partners in trust to protect the land. We approach our mission as if the world depends on us, because in many ways, it does. More soybeans are grown here than anywhere in the world. Roughly 80 million metric tons a year are raised on over 30 million hectares of land. As a result of sound science and continued research over the past 30 years, we have increased soybean yields by 55% on roughly the same amount of land. U.S. soybean farmers have reduced carbon emissions, energy use, greenhouse gas, and soil erosion. We're licensed to use pesticides, insured to protect workers' rights and safety, and our assurance programs are verified. We're meeting U.S. laws and international sustainability standards. And to validate our compliance, a team of 3,000 career conservation agents throughout the country and continual verification through surveys, interviews, field observations, and electronic data that we're operating our farms in compliance with stringent environmental laws. Our commitment is more than just a neighborly agreement. Independent peer-reviewed research verifies U.S. soy is sustainably produced, with comprehensive data demonstrating that U.S. producers are compliant with the vast majority of our hectares already subject to annual audit by the USDA. And because we are driven by a commitment to continuous improvement, we're incorporating soil testing, plant tissue analysis, satellite positioning that precisely adjusts crop inputs, and much more. Why? To ensure that every link in soybean production is sustainable and to live up to our commitment as stewards of the land. If a U.S. soybean farm could tell its own story, it would be a legend, complete with dedication, resilience, compassion, and above all, successful, sustainable perseverance that lives to tell the story all over again. United States Soy, verified sustainable. Learn more about our U.S. Soy Sustainability Pledge at ussec.org.
A very good afternoon to you all again. We are starting the wildlife and aquaculture session right now. We have 12 interesting presentations and hoping to have an interactive session with you all. And I encourage all our online participants to ask questions and get the maximum out of this session. I would like to introduce Dr. Ganga Vijay Singha as the session chair for today, Dr. Cecile Silva, Dr. Niranjala De Silva, and Dr. Shalika Pereira as our judges. Over to you, Dr. Ganga.
Good afternoon, everyone. All are welcome to the Wildlife and Aquatic session of the 73rd Annual Scientific Session of the Sri Lanka Veterinary Association. And there are 12 presentations, and there are some presentations on aquaculture, and there is a presentation on captive wildlife, and there are 2 presentations on free living wildlife annotators. So I would like to, the first presentation is on a case report of parasitic co-infection in goldfish, Carassius auratus, by HMM Madhushani, DM WCJ Disanayaka, and DPN De Silva. Over to you.
Good afternoon, everyone. I'm Prasad De Silva, and this is a case report of parasitic co-infection in goldfish. So, in aquaculture, infectious diseases are very common and cause great losses for the farmers and industry. Among these infectious diseases, parasitic infections, especially protozoan infections, are more common and abundant. So the objectives of this study was to diagnose the etiology of the disease in goldfish, which were found in our aquarium. And also, by presenting this, we would like to report the treatment protocol used to cure the affected fish. So first, we did the external examination by observing their clinical signs and also water quality parameters, to see whether there's any issue with water quality and all. And after initial treatments, after 2 days, we found that one fish with severe infection was dead, and we performed a post-mortem examination with the dead fish. So looking at the history, there were 3 adult goldfish around 6 years of age were found with symptoms of disease, and then we measured their length and weights, and they were around 16.7 centimeters in length and 55 grams, and they found mainly a deep lesion, like an erosion of the upper head near the sensory pits, which is also look like a hole in the head disease. So we wanted to investigate this further, and this erosion is deep in the dead fish, which I mentioned earlier. And also these fish showed a whitish fecal cast, and they were lethargic and anorexic. And also we observed that fin and tail rot, and in the dead fish it was very severe. So hemorrhagic patches near eyes, operculum, and base of the fins were also observed, along with one fish showing loss of balance. So this is the dead fish with severe erosion, like the lesion, ulcerated lesion of the head, and also observed severe fin rot. And this is an image of the white fecal cast hanging from the anus. And also one fish showed this loss of balance. And it also shows some fin rot here. So the external examination using wet mount preparations from skin, tail, base of the fin, and gills were examined under the microscope, and also the white fecal cast was also observed under wet mount. So, the results of this wet mount revealed that in the skin scraping, we found mild infestation with skin flukes, a Gyrodactylus species, and also the fecal matter showed the presence of flagellated protozoa called Hexameta species, and present it was called as pyronucleus, and then scrapings from the lesion of the head showed heavy infestation with flagellated protozoa Hexameta. So these are some wet mount images, preparations, and images of the skin fluke and Hexameta species that we have identified in all three fish species. So this video also shows the presence of live Hexameta on fecal cast. So we, as treatment, we had to combine several combination of treatments because it was having a co-infection with the skin fluke and Hexameta. So we gave a salt bath, a mild dose here for 3 days continuous, because the fish was sick and anorexic, and we didn't want to stress them further. And then metronidazole is the drug of choice, so we gave it as a bath treatment and repeated in every 3 days for over 3 weeks time. And we coupled this with vitamin C to relieve stress in fish. And also we added methylene blue bath for 3 days for fin rot, and also it acts as an antiprotozoal effect. And also we added, after 3 weeks, actually, there were no signs of external parasites. However, we observed the presence of Hexameta in the fecal matter of fish. So we decided to give an oral dosage of metronidazole, and they have regained their appetite by that time. So we gave orally metronidazole at a higher dose. And all the time we did a water change, 1/3 of water change in every 3 days. So this is during our treatment procedure, and you can see the presence of head lesion and still the loss of balance in fish. So after 2 days, actually, we found 1 dead fish, and we performed a post-mortem examination, and it revealed that the body organs were actually decomposed because it was a little bit late to observe the internal organs. However, the intestinal scosh or tissue scraping, we identified the presence of Hexameta species. So metronidazole is the drug of choice for hexametasis. So we used metronidazole for 4 weeks, and after 1 month, it was cured, and salt bath at low concentration was effective for controlling mild skin fluke infection. And methylene blue treatment was also act as a preventive measure to prevent secondary infections and also to treat fin rot and as well. And vitamin C was given to reduce stress, and it helped wound healing as well. So by week 1, fin rot, hemorrhagic patches near eyes, operculum, base of the fins, pectoral and caudal fins were reduced, and the absence of skin flukes by end of 2nd week. And at the 2nd week, eroded skin lesion, fins became normal, and week 3, no external Hexameta species were observed. However, the white fecal cast contained Hexameta species. So we performed oral dosage, and by the end of week 4, both goldfish survived, and they were free from hexameter species. So here you can see the fish before treatment, and they after the treatment they regain their color and the fins. So thank you very much for listening.
Thank you, Dr. Madhishani, for your interesting presentation and sticking under the allocated time. Now the paper is open for any questions. The virtual audience can use the raise hand option or can post their questions in the Q&A session or the chat box. Do you have any questions on this paper?
Yeah, it's a very nice presentation indeed. Thank you so much for that presentation. I just want to know that the hexameter species, there are several species involved in this genus as far as I know. So are all these hexameter species involved in causing disease in fish are of the same pathogenicity, or are there any difference in the pathogenicity? And do you have any guess as to what can be the specific species involved?
Yes, madam, thank you for the question. I hope you can hear me, madam.
Yes.
Yes.
Yeah.
So actually, there are different types of hexameter species, as you clearly mentioned, madam. In this, among these, actually, in fish, there are mainly 3 genera hexameter species. But we didn't in this study. Actually, we didn't go for the species identification. Hexameter intestinalis is the most common one, which is present in both GI tract. So it may affect other fish, and also not only freshwater, even marine fish also can be affected. I think I answered your question. Is it clear, madam?
Okay. And as far as you know, are there any predisposing factors causing the disease in fish?
Yes, madam. Mainly the water quality and also malnutrition are the predisposing factors. In addition to that, actually, co-infection with other parasites also may lead to stress and increase severity of infection with hexameter.
Thank you. Okay, Dr. De Silva.
Yes, madam.
You said this hexameter intestinalis affects the marine fish as well.
Yes, madam. I saw some reports of different hexameter species, especially salmonids, were affected with the hexameter species.
Okay, but they are in salt water. Doesn't that water help preventing the disease?
Maybe, madam, because they are inhabiting in the gut of the organism species, so it may have some other effects. The clinical signs may not be the same as in freshwater. Yeah, so they are in the gut. Mainly, they are intestinal parasite, but in certain instances, it may move. And during their life cycle, also they release to the water and then enters through the oral route.
Okay, does that mean the salt water doesn't have any effect on the parasite when they are released to water? I mean, in marine fish.
Yeah, madam. Yeah, they may be resistant because in the during transmission stage they make the cyst. So that cyst is maybe resistant to salt. And in our case, actually, we use salt not to treat the hexameter, but they had another skin fluke infestation. So that's why we applied salt as a treatment as well.
Okay, which is more severe, the skin form or the intestinal form? You said, I remember at the end of your, and it was the end of your treatment, the intestinal is still present.
Yes, madam. Yeah. Actually, we don't know whether it's we couldn't confirm the species Hexameta intestinalis but however, by looking at the morphology, it was confirmed as Hexameta species. So even though we at first we gave metronidazole, which is the drug of choice for hexametasis. So we gave it as a bath treatment at first 3 weeks, because the fish were anorexic by the time we start treatment. However, at the end of 3rd week, we found the parasite present in the fecal content. So we started giving the oral dosage. So after that, 1 week later, we checked the fecal smears and it was negative for hexameta. Yeah, so I think oral treatment should be more effective than the bath treatment.
Yeah, how do you make sure that the fish gets adequate amount of drug? Do you how do you give it orally?
Yes, madam. We actually dissolved it in water and using a syringe, we fed it into the mouth. In the presentation, there was a slide as feeding the oral route. Yeah. We wanted to make sure that it takes, otherwise we can mix with the food, but in here we wanted to make sure that the drug goes in. So we fed using a syringe.
Thank you.
Okay.
Can I add a comment to what Dr. Prasadi said?
Yes.
Yeah, like she said, the idea of using salt in this case is to prevent the secondary bacterial infections. Of course, we can use salt to treat hexameta, but the thing is, it depends on the species, because if we are to use salt as a treatment for hexameta, it should be a very high concentration. But we can't use it for certain species of fish, especially with the freshwater ones. But with the marine ones, perhaps because the hexameta is also living, the particular species is living in the sea, it's not susceptible for salt, then we have to go for other drugs like metronidazole.
Thank you.
Thank you, Dr. De Silva, and the participants and the judges who made this session very interactive. So we'll move on to the next presentation now. It's on growth, performance, and survival of Indian major carp, catla, post-larvae fed with different density of freshwater zooplankton, Daphnia magna. It is presented by P.A. Sanduni Madhushani, MNM Fauzi, and PM Vitanagi.
Good morning to everyone. We are going to present the project with the title of Effect of Partial Replacement of Concentrate Feed with Water Zooplankton Daphnia magna on growth, performance, and survival of Indian major carp, catla post-larvae. And our outline, we have designed our presentation with these titles, with the subtitles. So first, I'm going to introduce about this fish. The catla fish is one of the fast-growing Indian major carp, and it is an economically important freshwater food fish because they have a very high nutritional quality for human consumption. So it is one of a food fish. So generally the food fish prefer zooplankton as a live feed, especially for their early stages. We call them post-larvae. Daphnia magna is zooplankton, is a microcrustacean which contains high nutritive value, such as 20% or 20 to 27% of lipids, and they have up to 85% protein, which is a bit higher than the fish meal, and more amount of the enzymes and the minerals. So for our experiments, we designed hapa, so which is like cages. This is to provide the Daphnia. This is to provide the Daphnia as a food also as a feed to the early stage of the Catla, which is Catla postlarvae. So the net size of these cages, this HAPA, very important here to regulate the release of newly born Daphnia to the fish tanks. So the size of the cages, size of the net will facilitate, because the whole size will only allow the newly born Daphnia. So the adult Daphnia will retain inside the HAPA, therefore it will not be consumed by the fish. So therefore there will be a continuous production of this newly born Daphnia. Okay, this experiment was used to select appropriate density of Daphnia to feed Catla postlarvae. So the density of the Daphnia can be calculated easily by number of the cages or the HAPA placed in the fish tank. So the problem we identified in our before we start our project, our research, Catla is an exotic species which were introduced from India. So they are Indian carp, Indian major carp. They have a very good growth. So the research papers show that it has a very good, growth rate. But the problem is they have a low tolerance to environmental variation. When you bring to the unfavorable or they are different environments, they don't perform well. So in Sri Lankan water, we have identified they are, especially the early stages, very low survival rate, and the growth rate also less comparatively. So we then thought of, it was suggested by many other species. So Daphnia magna is a zooplankton, can be introduced to this live feed, as a live feed to this Catla. But there was no definite procedure yet in Sri Lanka, and there is no specific method to get an optimum growth performance of Catla. Therefore, our objective of our research was to design the cage or HAPA for releasing the newly born Daphnia magna to release into the fish tanks. And the second objective is to determine the growth performance and the survival of Catla by feeding newly born Daphnia magna. And the third objective is to determine the density of Daphnia magna to replace the concentrate feed for optimum growth performance of Catla postlarvae. So this is the very summarized or the methodology that we have followed. First, we prepared the pond of the fish. That is the concrete fish pond, okay, cement tank. Then we stocked the fish. These fish were directly got from the breeding or nursery. Then beforehand, we have prepared, we have brought the Daphnia stock and we have cultured as a master culture. And we have also prepared the HAPA, okay, and we were prepared 9 cages of HAPA. So we followed 3 weeks. First week, we did not give a Daphnia. Okay, so we have just these are acclimatization period. We have given as a usual feed for the fish. That is first week we soya milk and other concentrate feed. The second week, in addition to this, we introduced the Daphnia with a high percentage. Okay, 100,000 Daphnia per 1 HAPA. So in the third week, we have topped up with another 50,000. So this is a unique stocking rate of Daphnia for each cages. So afterwards, we harvested the fry or the larval fish. Then we analyzed the data. Okay, let's go to little deeper into the methodology. So we have purchased, we have collected the Daphnia sample from ornamental fish center at Rambodagalla, where we brought to the experimental stations, which is a nectar breeding station in Murupavala. where the Daphnia were cultured in a mass culture in the cement tank. Then at the same time, around 50,000 catula fish were brought and acclimatized, and 5,000 catula post-larvae were stocked in each of the 9 experimental tanks after the acclimatization. So the stocking rate were maintained for all of the tank as 300 post-larvae per square meter. And the hapa which were supposed to be fed to the supposed to be given to the fish tanks were stocked in the cages, that is the hapa. 9 cages also, because 9 tanks, fish tanks we used. For each tanks we were used this for differently. I will show you how we have introduced. And then the pond was selected and prepared for experiment. Then the data collection such as length of the fish, the weight, initial weight, and every week we have collected the data. And the survival also we have collected. So finally the data were analyzed by using the Minitab 16 software by one-way ANOVA test. So this is the experimental design. We had 3 treatments, each with 3 replicates. Treatment 1 was used as a control where we did not give a death. But the treatment 2, where we have introduced for each tank of the treatment 2, we introduced 1 hapa. Where the first week we did not introduce the Daphnia, but the second week we have introduced 100,000, we stocked 100,000 Daphnia per 1 hapa. But the third week we have topped up another 50,000. So similarly, for the tanks, fish tanks of the treatment 3, so where there were 3 tanks, and for each of them we have introduced 2 hapa. So this is the difference between the treatment 2 and 3. 2 hapa we have introduced. So therefore, finally, the second week we stocked 200,000 Daphnia, mother Daphnia. Okay, and the third week we topped up another 50,000. Therefore, this is another the 2 hapa. Therefore, 100,000. So totally for these 2 weeks, second and the third week, we introduced 300,000. Whereas the treatment 1, we introduced only 150,000. So this is our experimental experiment design. So whereas other concentrate feed and that is a commercial feed and soya meat were common for all of them. So let's see our results. So our results was very interesting to see. The growth has significantly increased in the treatment 3, in the tanks of the treatment 3, where we stocked 300,000 Daphnia in the hapa in the last 2 weeks. So where we have found the final growth, final length we have measured significantly increased than other treatment. This the control one is 24 millimeter, but whereas the treatment 3 was 35. In the same manner, the final weight also has increased almost higher, because their width might have increased more. So the length is not double, but the weight was 4 times higher than their initial the control weight. And we have also recorded the length gain. So every week we have taken, every 3 days we have taken the sample. So in every 3 days we have seen the significantly improved, increased the mean total weight. So in the 21st day we have seen the highest. Okay. Similarly, the total length also has increased significantly. And the survival rate of the catla also has increased. So in the treatment 3, the survival rate was 78.7%, so which is almost double of the control. So the survival has significantly, because we thought this could be due to the many reasons that I will discuss at the discussion. But in the briefly to tell that the live feed may have minerals and the vitamins, which will and also some high protein content, which will increase the immunity of the fish. And the specific growth rate also in the treatment 3 has increased significantly. So according to the experiment, our Daphnia, by providing the Daphnia in 2 cages, has increased the growth performance of the cattle and reduced the mortality rate. And similar results have been recorded in a very recent study. In 2021, this was published in the other species, not the same species, mullet fish. They have replaced 75 to 100% partial substitution of the fish meal with the Daphnia magna meal. So they have made this, we have given Daphnia magna as a live feed, but they have prepared the meal out of this Daphnia magna, but they have also shown the improved growth performance and feed utilization. So the cages, we have discussed here that the cages has facilitated the slowly release of the newly born Daphnia to the fish tanks, where the size of Daphnia is suitable for the mouth of the post larvae. So therefore, it was facilitated the adult Daphnia breeding inside, while the continuous supply of the newly born Daphnia to the larvae, to the fish. So this is the adult Daphnia, which was stocked in a hopper in 2 regimes, in the 2 weeks, 2nd week and the 3rd week, we have supplied. So it is a supplementation. So that's a top up. So it is also benefited. So the reason of that is adult Daphnia is performed high reproductive performance in the 1st week. And after they become mature, their life is, since the life cycle is short, they may die. So therefore, we need to top up. So that is the reason why we have top up. This is also we have got this back from the other published data. So therefore, we got this idea and we have used this. This was very beneficial in our research. So as a conclusion of our study, the feeding the cattle post larvae with the Daphnia has increased the growth and the survival rate and reduced the mortality rate of the cattle post larvae. So furthermore, the feeding the cattle post larvae with 300,000 adult Daphnia evenly in 2 cages has resulted highest growth and the survival rate compared to 150, the half of that. Okay, 150,000, that is the 1 cage. So this is very, according to our research, we have found this is very effective. But the releasing Daphnia magna by using the hopper to fish of cattle is better than introducing Daphnia directly to the tanks. But here one more, the latest, after our published, I have found another research that they have made the fish meal, the Daphnia meal, which has increased much more. So therefore, the knowledge has to be, this experiment has to be studied some more details with more treatments, because we have found if you increase their growth may increase. Therefore, this cannot be, we can't say that this is the final density of the Daphnia that can be given, but definitely we had to do some more experiment to come to the conclusion that it can be, but we can say definitely that Daphnia magna has definitely increased the growth rate and reduced the survival rate. So we have referred many articles up to date, and thank you very much for.
Thank you, Dr. Fauci. And I was told that Dr. Madhushani will be joining us for the Q&A session. So this paper is open for questions. Any questions from the audience or virtual participants or the judges? Go ahead, Dr. Sujay.
Thank you very much. I think it's very important practical study. Do you have the nutritional profile of Daphnia? And if I'm right, your concentrate feed is same for all three groups, and your level of Daphnia is high. It's like a dose response trial. And definitely for your control, your treatments get more nutrition than your control. Then you would expect your treatments do better than your control. Do you have any explanation for that?
Yes, sir. Catla PL most is preferred to hunting. Catla postlarvae prefer to hunting. The Daphnia has some movement, and therefore Daphnia postlarvae like to bite Daphnia than concentrate feed.
But by giving Daphnia, you're not substituting your concentrate feed. So they get the same amount of concentrate feed. On top of that, your treatment 2 get X number of Daphnia, and treatment 3 gets even more. Then you would expect treatment 3 to do better than 2 and than the control. Do you have any explanation, or do you get say much more than the nutrient you provide?
The treatment uses a control.
Yeah, control has certain level of concentrate. The treatment one also has the same amount of control concentrate. Is that right?
Yes, sir.
Yes. And then you get something extra for your treatment. And then definitely they have a better nutrient intake or more nutrients in treatments than your control. Then your comparison becomes little difficult.
Dr. Madhusani, are you with us?
Yes, sir. Yes, ma'am.
Did you get the question?
Yeah, did you understand my question?
Yes, sir.
Yeah. So you have a control group with, I don't know, maybe to satisfy the nutrient requirement, you're feeding them. And then another group, on top of that, you feed something else. Is it much more nutrient?
Same concentrate feed use every treatment.
Yes. But you give Daphnia in addition to that. So treatment not to your control. Whatever nutrients, say, I don't know the nutritional profile of Daphnia. So your treatment groups get more nutrients than your control group. Second, third one, or the treatment 3 get even more. Then someone would expect treatment 3 to do better than 2 and 2 better than the control. Any explanation for that?
Dr. Madhusani?
Yes, ma'am.
Okay, then we'll move on to the other. We have got questions from the virtual participants. what would be the lifespan of Daphnia adult?
Sorry, ma'am.
That's a question asking what would be the lifespan of Daphnia adult? Lifespan of Daphnia.
Say, in other words, how long you should change the adult stock?
Daphnia, first week only, they performs high maturity rate, high reproduction.
So, at what age of adult, the age of that adult Daphnia? What is the age you prefer?
After maturity, 5 days only use reproduction. Maturity become newly born after first week. Then maturity, we should use second week only reproduction for the adult Daphnia. Therefore, we should introduce the Daphnia into the Hapa.
Yeah. So when you reintroduce, replace the stock, the earlier stock is there, no? You don't remove them?
Early Hapa.
Yeah, earlier adult was there. They were there.
No, someone is dead and someone weak reproduction.
Okay, thank you, Dr. Madhushani. And due to, I'm sorry, due to time constraints, we have to move on. So the next presentation is on systemic aspergillosis in a captive newly hatched emu chicks, Dromaeus novi hollandiae. The by P. Ejiwana, I. Ratnayaka, A. Palkumura, T. Gunawardana, T. Rajapaksha, R. Jinadasa, and K. Vijayasundara. And this will be presented by Dr. K. Vijayasundara.
Hello everyone, I am Dr. Prasad from Ridigama Safari Park. I am working as a government veterinarian there. It's under National Zoological Department. I'll be going through a case of systemic aspergillosis in a captive newly hatched emu chick. So first of all, I'm talking about who is emu. It's second largest living bird by height after the ostrich. It's endemic to Australia and they're actually omnivores and they are doing their normal behavior things during daytime. So we call them as diurnal animals. They are most common and valuable zoo animal. There is a huge demand globally and only found in zoological gardens and breeding birds park in Sri Lanka. Then I'm talking about aspergillosis. Actually, it's a pathogen for birds. It's a fungal infection that commonly causes respiratory disease in pet birds. It can cause both upper and lower parts of the respiratory tract and causing respiratory problems, or more broadly distributed systemic infection like peritonitis. Aspergillus fumigatus is the most common one. It's causing 95% of cases, and the second most common one is Aspergillus flavus. It's the second most common one. It's very important to know what are the predisposing factors because treatment is very difficult, and when we treat them, they may get stressed, and it's very difficult to manage them under the treatment. They may get stressed and they may be anorexic. So it's better to take the precautions by knowing predisposing factors. Warm and humid environments So combined all three together and kept it under 30 centigrade temperature whole day. It showed signs of difficulty in breathing one day. Then it was taken to the hospital and treated with antibiotics and other supportive drugs. After the treatment, it was a bit improved, but few days after, it was found recurrent and died during the treatments. All the signs were related with respiratory disease. The carcass was sent to the Department of Veterinary Pathobiology, University of Peradeniya for further investigations. A detailed postmortem examination and other lab techniques were performed, confirmed that it was severely infected by Aspergillus flavus with histological and pathobiological evidences. Here we have gross pathological findings, pale conjunctival and oral mucosa. It was anorexic couple of days before the death, and multifocal gray to yellow iris. Irregular nodules were in lungs, air sacs, and peritoneum. Additionally, lungs and air sacs appeared congested and edematous. Samples were collected for histopathology and microbiology for further investigations. Here you can see the multifocal gray to yellow nodules in the lung parenchyma and peritoneum and air sacs. Laboratory techniques, samples were fixed in 10% neutral buffered formalin and underwent routine tissue processing. Formalin fixed tissues were used for HE and fast staining. Lung sample was cultured blood agar and subarachnoid dextrose agar and incubated aerobically at 25 centigrade temperature. Histopathological findings, formalin fixed wax embedded tissues were sections at 2 micrometer thickness and stained with hematoxylin and using an periodic acid skip. Variable sized granulomas were observed in HE stem lung parenchyma, like we have seen so many nodules at the lung parenchyma in gross pathology. Developing fungal hyphae with acute angles in branching were seen in past stain section. Here you can see variable sized granulomas in the lung parenchyma in histopathology. Then developing fungal hyphae with acute angles in branching under the fast stain. Microbiological findings, there was no any aerobic bacterial culture. The sample was negative for aerobic bacterial culture. Only the cotinimycelium appeared first on subarachnoid dextrose agar and changed the color to yellow green later. Wet mount stain with lactophenol cotton blue stain showed characteristic fruiting head appearance of Aspergillus species. Round vesicles with sporulation over the entire surface suggestive Aspergillus flavus infection. The final suggestion is aspergillosis should be considered as a major differential diagnosis in respiratory diseases of young captive wild birds in Sri Lanka, because they are very valuable. They have huge demand globally. If you can save them, save one animal, it will be very economical for the country also, and the world will recognize us as a very good breeding centers. So it's very important to keep aspergillosis as a major differential diagnosis when they get respiratory diseases, when they are at their very beginning, very early stages of their life. Thank you very much. And if you have any questions and clarification, please ask me. Please let me know here for
Yes, are you with us? He's there, but can you hear?
Can you hear us? Dr. Prasad, can you hear?
I can hear you.
Are you muted? We can't hear you. Prasad? Prasad, we can't hear you. Can you unmute and check? It seems like you have issue with the microphone. Yeah. So it seems like you have an issue with the microphone. We can't hear anything you say. So I'm sorry, we have to move on to the next paper. Thank you, Dr. Prasad, for your interesting presentation. And the next presentation is on high occurrence of multi-drug resistance among gram-negative bacterial pathogens in freshwater ornamental fish by DSPM Danapala, RS Kalupahana, AW Kalupahana, DPH Vijayasekara, SA Kotavatta, NK Jayasekara, A Silva Fletcher, SSSDS Jagoda. Dr. Jagoda will be presenting the paper.
Hello everyone, today I am going to present our study titled High Occurrence of Multi-Drug Resistance among Gram-Negative Bacterial Pathogens in Freshwater Ornamental Fish. Introduction. Ornamental fish industry in Sri Lanka is rapidly growing and has a great potential for expansion. It supports the country's economy through foreign exchange earnings, and Sri Lanka is having 4.2% share in global market. Main fish varieties exported from Sri Lanka are guppy, neon tetra, platy, swordtails, and molly. Bacterial infections in ornamental fish. It is one of the most common health conditions in tropical freshwater fish. It can be either systemic or external infections. These bacterial infections are mostly caused by gram-negative opportunistic pathogens such as Aeromonas species, Edwardsella species, Pseudomonas species, and Citrobacter species. These bacteria can cause hemorrhages, fin rot, skin ulcers, exophthalmia, and distended abdomen in fish. Use of antimicrobials in aquaculture. Antimicrobials are used both therapeutically and prophylactically to control bacterial diseases. And commonly used antimicrobials include oxytetracycline, erythromycin, endofloxacin, amoxicillin, chloramphenicol, and sulfamethoxazole trimethoprim. There are some concerns while using antimicrobials in aquaculture sector, such as use of antimicrobials without proper veterinary intervention, prophylactic use of antimicrobials on apparently healthy fish, release of medicated water and effluent water to natural environment. This long-term misuse of antimicrobials has resulted in the development of drug resistance in aquatic bacteria. This has led in difficulties to treat infections in fish and spread of antimicrobial resistance by horizontal gene transfer, which is a public health risk. Our objectives of this study were to characterize gram-negative bacteria isolated from diseased freshwater ornamental fish and to evaluate the antimicrobial susceptibility of isolated bacterial pathogens. Now let's move on to the materials and methods. Collection of samples. Ornamental fish which were showing septicemic signs were collected from ornamental fish breeding farms and aquaria, and also samples which were submitted to Center for Aquatic Animal Disease Diagnosis and Research for disease investigations also were used. Swabs from ulcers were collected from the fish with external bacterial infections, while necropsy was performed to the fish with generalized septicemia, and kidney were collected aseptically. Both of these samples were cultured on Triptyxo agar and incubated at room temperature for 24 to 48 hours. After the incubation period, isolated colonies were used for gram staining, and gram-negative bacteria were used for preliminary biochemical identification using oxidase, catalase, and oxidation and fermentation tests. Then genetic characterization and antimicrobial susceptibility testing was done. DNA was extracted from isolated colonies and Guide gene amplification was done. Then those sequences were analyzed for species identification. Antimicrobial susceptibility testing was done according to the CLSI guidelines. We used 13 antimicrobials, including amoxicillin, nalidixic acid, rifampicin, chloramphenicol, doxycycline, erythromycin, enrofloxacin, tetracycline, sulfamethoxyl trimethoprim, nitrofurantoin, gentamicin, ceftazidime, and imipenem. In here, you can see the primers and thermal profile, which were used for Guide gene amplification. Now let's move on to the results. We could isolate 42 gram-negative, oxidase-positive, fermentative bacteria from 29 fish that belong to 8 different species. In here, you can see some ornamental fish breeding farms, and this is a mud pond, while these are cement tanks. In this slide, we have shown some fish with clinical signs of septicemia. Here, this one is having hemorrhages, and this is a fish with dropsy, and this one is having ulcers, while this fish is having hemorrhages as well as fin rot. These are some culture plates of primary cultures, and here you can see isolated colonies. This is a gel image of Guide gene amplification. In this graph, you can see the bacteria we could isolate from our study according to the Guide partial gene sequence analysis. As you can see, we could isolate Aeromonas veroni, Aeromonas hydrophila, Aeromonas jandei, Aeromonas darkensis, Citrobacter frundi, Morganella morgani, Enterobacter rogencampi, Shevanella decolorationis, Phytobacter diazotrophicus, and Quasimonas shigelloides. In this graph, you can see the antimicrobial resistance of gram-negative bacterial isolates as a percentage, and the highest resistance has shown against amoxicillin, which has been followed by enrofloxacin, nalidixic acid, tetracycline, and erythromycin. Isolated bacteria showed considerable resistance against amoxicillin, enrofloxacin, erythromycin, and tetracycline. Of all isolates, 30.7% were multi-drug resistant, and 36.7% of multi-drug resistant bacteria showed resistance to 5 or more than 5 antimicrobial classes, with one isolate being resistant to 8 antimicrobial classes. When multiple antibiotic resistant index is considered, it is a quite useful tool to risk assessment by identifying antimicrobial contamination from high-risk environment. In our study, this MAR index was ranged between 0.08 to 0.77, and 78.6% isolates were showed MAR index, which is higher than 0.2. MAR index higher than 0.2 suggests that they originate from high-risk source of contamination where antimicrobials are often used. Discussion. Aeromonas species were predominant gram-negative bacteria isolated from diseased ornamental fish. A. veroni, A. hydrophila, Aeromonas jande, Aeromonas darkensis, Citrobacter frundi, and Fleishiomonas shigelloids are opportunistic pathogens reported to infect ornamental fish. However, the pathogenic role of rarely isolated gram-negative bacterial species such as Phytobacter diacetrophicus, Shewanella decolorationis, Morganella morgani, as fish pathogens is not properly understood and should be investigated. High levels of resistance was observed against antimicrobials, which are frequently used in aquaculture. Example, tetracycline, enrofloxacin, erythromycin, amoxicillin, sulfamethoxazole, trimethoprim. And the least resistance was observed against ceftazidime, which is a third-generation cephalosporin, and it is rarely used in aquaculture. Comparatively high resistance against gentamicin and imipenemys elami, as these are not usually used in treating fish, but they use in treating humans as last resort of antimicrobials. Conclusion. Bacterial diseases in ornamental fish can be controlled successfully with antimicrobials and environmental improvement if diagnosed early in the course of the disease. Ornamental fish and their carriage water can act as reservoirs of MDR bacteria and therefore pose a public health risk. High occurrence of multi-drug resistance among gram-negative bacterial pathogens in freshwater ornamental fish highlights growing need of veterinary intervention to safeguard this thriving industry. Acknowledgement. We would like to thank freshwater ornamental fish farmers, hobbyists, and aquarium owners, academic and non-academic staff at Centre for Aquatic Animal Disease Diagnosis and Research, UKRI Research England for their financial support. These are our references. Thank you.
Thank you, Dr. Anapala, for your eye-opening presentation. Now the paper is open for questions. I have one question. In aquaculture, do they use veterinary products or human products? The commonly, you mentioned some commonly used antibiotics. Are you with us? Is any of the authors joining with us online?
Dr. Ganga, can you hear me?
Yes, we can.
Actually, they use human products. There are no veterinary products as such in the case of antimicrobials. But antiparasitics and other chemicals, there are aquatic products meant for ornamental fish use. But in the case of antibiotics, they use human products.
So, when without any veterinary intervention, is it?
Yeah, that's what's happening in the field.
Okay. Okay. Dr. Samantika?
Yes, madam.
Yeah. Now this, how do they dispose this water?
Actually, madam, very few ornamental farms have their own septic pit to which they discard water, but many of them we know discard treated water as well as the naturally discarded water to the environment, to the natural water systems.
It will have a very bad impact on the environment, isn't it?
Madam, can I comment on this?
Yes.
Yeah. Since we deal with most of the ornamental fish farmers, most of them are advised to, before discarding the water, they treat the water with the disinfectant. And some are, having the pits and the drained water is undergoing the purification methods. But some, of course, not. But they are educated farmers who do the treatment and, then discard the water. Thank you.
Thank you. Samantika, can I ask another question regarding this effluent? All these water treatments, will there be any effect on the concentration of antibiotics if it contains antibiotic residues? And do they have to get any approval from the Central Environmental Authority or some organization for aquaculture and discharge of water, wastewater?
Actually, large-scale farms may have their own arrangements, but the small-scale and middle-scale farmers, actually, we have visited many of these farms to take samples because what Dr. Dhanapala has presented is a part of an ongoing research on antimicrobial resistance in aquatic environment. So we collected samples from biofilms, effluent water, pond sediment, and many more samples were taken for this study. And we observed that many of them just drain the water to the outside of the farm without treatment. Maybe large-scale farmers may have their own arrangements.
Okay, thank you, Samantha. Are there any more questions? Thank you for your presentation. So we'll move on to the next presentation. The parasitic dinoflagellate hematodinium species as a cause of heavy mortality cultured mud crab Cicella serrata in Sri Lanka. It is presented by RMIM Ratnayaka, RNPK Ranatunga, AW Kalupahana, GST DS Gunavardhana, and SSS DS Jagoda.
Hello everyone. I am presenting about parasitic dinoflagellate hematodinium species as a cause of heavy mortality in cultured mud crab Cicella serrata in Sri Lanka. Mud crab is a popular aquaculture species worldwide. There is a high demand for Sri Lankan mud crabs in foreign countries. Supply of mud crabs in Sri Lanka mainly depends on fattening of wild-caught soft-shelled crabs. Commercial mud crab culture is still in its infancy in Sri Lanka. Mud crab is a promising species for diversification of coastal aquaculture. An outbreak of disease was reported from November to December in 2020 in several areas in northwestern province of the country. After stocking hatchery-bred crablets in mud ponds, high mortality of about 60 to 100% was reported in sub-adult to adult crabs of 2 to 7 months of age. Deceased crab samples were submitted to Center for Aquatic Animal Disease Diagnosis and Research by National Aquaculture Development Authority. The objective of our study was to investigate the causative agent of the disease outbreak. The investigation was based on several criteria. They were external examination and necropsy, bacteriological investigations, histopathological examination, and molecular identification. For external examination and necropsy, crabs were anesthetized by chilling in 4 degrees Celsius on ice. Hemolymph was collected onto a glass slide and fixed with buoyant solution and stained with hemotoxylin and eosine and observed under light microscope. During external examination, we looked for changes of the color and texture of carapace, presence and absence of epibiond fibrillin and limb loss. Wet mounds of gill clips and the scrapings of carapace were observed under light microscope. For bacteriology, hemolymph was cultured on tryptic soy agar with 3% salt, and TCBS agar. The plates were incubated aerobically at 25 degrees Celsius for 24 to 48 hours. For histopathological examinations, hepatopancreas, gills, heart, and muscles were fixed in Davidson's fixative and were processed routinely using paraffin histological techniques and stained with hematoxylin and eosine and observed under light microscope. For molecular identification, DNA was extracted from hepatopancreas, hemolymph, and muscle using Quagen DNA mini kit, and conventional PCR was performed to amplify a region of the hematodinium 18S rRNA gene using hematodinium specific primers. When it comes to results, we received samples from 3 affected farms in Putlam district, and 8 live mud crabs were transported to the laboratory. However, 5 of them have died during transportation, and we proceeded with 3 live crabs. During the external examination, we observed lethargic behavior, dull carapace with varying degrees of hyperpigmentation and absence of one or several peripods. This is an image of uninfected crab and infected crab dorsal surface. The carapace of the infected crab shows the darkening of several places and absence of periopods. This is the ventral surface of the uninfected crab and infected crab. During hemolymph analysis, hematodinium infected mud crabs showed opaque and cloudy white hemolymph compared to the transparent hemolymph of the uninfected crab. Hematodinium-like uninucleate parasites were present in large numbers of the hematoxylin and eosine stained slide with host hemocytes in less number. In gross pathological findings, the uninfected crab's ventral side of the carapace shows a pooling of clear hemolymph. This is the infected crab. Pale gills and pale soft internal organs that appear frosted with a friable white residue can be observed in the infected crab. No significant bacterial pathogen was isolated in the bacteriological investigations. This is a photomicrograph of the hepatopancreas. Infiltration of large numbers of hematodinium parasites in hemal spaces can be observed here in low magnification. This is the higher magnification of the same image. Heavy parasitic infiltration in the hemal spaces can be observed here too. In molecular investigations, the anticipated hematodinium 18S rRNA gene-specific 187 base pair amplification product was successfully amplified from DNA samples of hemolymph of all 3 crabs. In addition, hepatopancreas and muscle of heavily infected crabs yielded the expected amplicon. This is the gel image, which shows the 187 base pair band. When considering our results, based on the history, gross pathology, histopathology, and molecular findings, It confirms that Hematodinium species is associated with this outbreak. Endoparasitic dinoflagellates in the genus Hematodinium are significant pathogens affecting the global decapod crustacean fishery. Mortality associated with this disease outbreak caused a large economic loss to the farmers who naively invested on commercial culture of mud crabs. Management of Hematodinium in diseased mud crabs remains challenging as there is no proven treatment measure. Further investigations on the causative agent and epidemiology are required for the future sustainability of the emerging mud crab industry. In conclusion, preventive measures to minimize the disease burden due to Hematodinium species should be taken. Removal of infected crabs from stocks during earlier stages of infection, proper disposal of infected crabs, screening of wild-caught broodstock prior to using breeding are some of the measures that can be taken. These are some of our references. We are very much grateful to Dr. A. Arulkantan for his constructive inputs, and we are also thankful for these personnels mentioned. Thank you all for your attention.
Thank you, Dr. Ratnayaka. It's a very interesting presentation. So the paper is now open for questions.
Thank you for the interesting presentation. And just for my curiosity, is this the first time that this has been reported in Sri Lanka?
Dr. Ratnayaka, are you there?
If she is not there, I can answer that question. Is that okay?
Yes, Savantika, go ahead.
Yeah, according to the published literature, there have been no reports of Hematodinium from Sri Lanka.
So this is the first time?
Yes.
Okay. And another thing is there any effect of salinity on the survival of this organism?
Actually, we are still in the middle of investigations. According to what we heard from the farmers is that when the outbreaks were occurring, the salinity went down. Actually, the usual salinity in this the crab farming, what they expect is around 32, 33 ppt, but when this incident happened, the salinity went down up to 16. That was an after effect of that cyclone Burevi, because there were a lot of rains and the seawater diluted. That can be one reason, because in other countries, hematodiniasis has been reported along with this salinity drops.
Okay. So you check the salinity in this study?
Actually, we got just one water sample, and in that also the salinity was lower than the expected value.
Okay, thank you.
Samantika, do you have any idea about the importance of significance of this Hematodinium species in wild mud crabs? Are there any reports of outbreaks in anywhere in the world?
Yes, Dr. Ganga, there are a lot of reports about hematodiniasis in wild crustacean species, including wild crabs, but we have not done any study. In this outbreak also, we think that it introduced from wild broodstock.
Thank you.
Ganga, is there any more time for more questions?
Yeah, you can have,
Yeah.
you can answer one question.
Yeah, thank you, Dr. Samantika and your team for this interesting presentation. So were there any history of toxic algal blooms in these water bodies where you got those crabs?
Thank you, Dr. Erandi, for the comments. Actually, we received this sample from National Aquaculture Development Authority of Sri Lanka for disease investigation. So we didn't go to the site and do an investigation. According to what we heard from them, there had been no such history.
Because there's in literature, sometimes the same hematodinium is associated with, there are free living stages reported. So if there's an associated history of a toxic algal bloom, maybe it can be the cause, I mean, the origin of this dinoflagellate.
Yeah, thank you very much. Yeah.
Thank you, Dr. and Dr. Ratnayaka and Dr. Erandi and other participants. So we'll move on to the next presentation. It's on detection of infectious spleen and kidney necrosis virus infection in freshwater ornamental fish from two provinces of Sri Lanka by DMPM Dhanapala, RMIM Ratnayaka, DMSD Disanayaka, RRMKK Vijayasundara, AW Kalupahana, and SSSDS Jagoda.
Hello everyone, I am going to present our study titled Detection of Infectious Spleen and Kidney Necrosis Virus Infection in Freshwater Ornamental Fish from two provinces of Sri Lanka. Introduction. Ornamental fish industry in Sri Lanka is rapidly growing profitable business. It contributes to the economy of the country through foreign exchange earnings. Sri Lanka exports farm-bred tropical freshwater fish and wild-caught marine and brackish water fish. And diseases, either infectious or non-infectious, have become a major challenge to the development of the industry. One of the viral infections in ornamental fish is megalocytoviral infection. It is caused by the genus megalocytovirus of family Iridoviridae. They are large double-stranded DNA viruses. And this megalocytovirus can cause severe systemic disease with moderate to high mortality in many species of freshwater and marine ornamental fish. As there is no effective treatment to control this disease, depopulation followed by disinfection is recommended. Now let's briefly look at what is infectious spleen and kidney necrosis virus. Family Iridoviridae contain 5 genera, including megalocytovirus. And megalocytovirus includes 3 groups, namely red sea dream iridovirus, infectious spleen and kidney necrosis virus, and turbot reddish body iridovirus. Infectious spleen and kidney necrosis viral infection was mainly restricted to Asian countries such as China, Singapore, Taiwan, and Korea. But at present, it has been reported in USA, Japan, Australia, and Germany also. Many freshwater ornamental fish are susceptible to infectious spleen and kidney necrosis virus. For example, cichlids such as angelfish, livebearers, and gourami. And this infection can be symptomatic or asymptomatic. If it is symptomatic, it shows clinical signs of lethargy, loss of appetite, abnormal swimming, distended body cavity, ulceration, hemorrhages, pale gills, and heavy mortality. Our objective of this study was to detect the presence of infectious spleen and kidney necrosis virus in freshwater ornamental fish stocks that experienced heavy mortality in Sri Lanka. Now let's go through the materials and methods. Ornamental fish samples with a history of heavy mortality were collected from ornamental fish breeding farms and aquaria. And the samples submitted to Center for Aquatic Animal Disease Diagnosis and Research with the complaint of heavy mortality was also used. External examination was done by observing wet mounts of skin scrapings and gill clips under the microscope. It was followed by the necropsy, and the kidney tissue were cultured on tryptic soy agar and incubated at room temperature for 24 to 48 hours. Gill's kidney, liver, and spleen tissue were fixed in 10% neutral buffered formalin for histopathology. And those tissues were used for DNA extraction, and molecular identification was done by PCR, which was followed by sequence analysis. In here, you can see the primer sequences we used for the amplification of megalocytovirus major capsid protein gene. And these are universal primers for all megalocytoviruses. In here, you can see the thermal profile we used. Now let's move on to the results. Ornamental fish from a total of 25 disease incidences were sampled. It included 11 from Western Province, 10 from Central Province, and 4 from North Central Province. Out of those 25, 8 fish samples gave positive results by PCR. Those samples included swordtail, angelfish, giant gourami, goldfish, zebrafish, and fighterfish. Diagnostic findings of infected fish. All those sampled fish showed clinical signs of anorexia, lethargy, darkening, erratic swimming, and abdominal distension. And there were no external parasites shown at the external examination. Bacteriological investigations revealed no significant bacterial growth except in one incidence where fish were also infected with Mycobacterium species. And here, hypertrophic cells and lymphocytic infiltration was seen in the histopathological examination. Molecular identification using PCR followed by sequence analysis revealed positive PCR with megalocytovirus universal primers. This is a gel image of amplification of major capsid protein gene by PCR using universal primers for megalocytoviruses. The positive samples has shown a amplicon at 777 base pairs. This is another gel image of the universal primer gene amplification. And in here, only one sample has given a positive results, and it has been isolated from an angelfish from Western Province. Sequence analysis. Aligning of partial gene sequences and comparing them with the sequences in GenBank database using blast service revealed the infection of infectious spleen and kidney necrosis virus. Our isolates had 99.8% to 100% homology with infectious spleen and kidney necrosis virus isolate A2, which is having the GenBank accession number of KP292956.1. Histopathological findings revealed hypertrophic cells, lymphocytic infiltration, and single-cell necrosis in liver and kidney. Moderate number of hypertrophied cells were present adjacent to the renal tubules. In here, you can see the histopathological findings. This is a hypertrophied cell in kidney, and this is a hypertrophic cell in liver. And in here, you can see lymphocytic infiltration in kidney. And here, this is the lymphocytic infiltration in liver. Discussion. Our study revealed the presence of infectious spleen and kidney necrosis virus in 6 different species of ornamental fish. And this is an emerging viral disease associated with high mortality and trade impacts, though it is not an OIE listed fish disease. Largely unregulated nature of fish movements between farms might have favored the transmission of the virus. Although there is no known report of infectious spleen and kidney necrosis viral infection in ornamental fish in Sri Lanka, previously it has been detected in ornamental fish imported to Australia from Singapore, Sri Lanka, and Malaysia. And also there is one report of the presence of infectious spleen and kidney necrosis virus in Sri Lankan sea bass, which was confirmed by histopathological findings. Conclusion. Our findings prove that infectious spleen and kidney necrosis viral infection persists in ornamental fish cultured in Sri Lanka. More systematic disease surveillance programs would be needed to understand the true prevalence and geographical distribution of this infection. Farmers, aquaculturists, and veterinarians should be made aware of this emerging disease. Acknowledgement. We would like to thank Dr. A. Arul Kantan, Senior Lecturer, Department of Veterinary Pathobiology, for providing the positive control, and ornamental fish breeders, farmers, and aquaria owners, academic and non-academic staff at the Center for Aquatic Animal Disease Diagnosis and Research and Department of Veterinary Pathobiology. University Research Grant, URG 2016-66V for financial support. These are our references. Thank you.
Thank you, Dr. Dhanapala, for your interesting presentation. Now the paper is open for questions. Yes, Dr. Roshan.
Yes, it's nice presentation. I mean, similar character. So I would like to know, how did you confirm the sequence? Once you got the sequence, how did you check the quality of DNA using any softwares, or how did you do that? Because once you sequence, you get a file. Once you get a file, how did you confirm the quality of sequences?
Dr. Roshan?
Yes, Samantika, go ahead.
Can you hear me? Yeah,
Yes.
actually, we sequenced this 777 BP fragment of major capsid protein gene of megalocytovirus. Then we aligned both sequences. The AB files, we aligned sequences both forward and reverse, and we edited by looking at the chromatogram. Then we submitted the sequences to GeneBank, and using the BLAST, we compared our sequence with all available megalocytovirus sequences in the GeneBank, and found that our isolates have homology of 99.8 to 100% to infectious spleen and kidney necrosis virus isolates from Korea.
Thanks.
Are there any more questions? Okay. Thank you, Dr. Dhanapala, Dr. Samantika, and Dr. Shan. We'll move on to the next paper. It's on mortality, rescue, and release statistics of Panthera pardus scotia in central hills of Sri Lanka during 2019 and 2020. Due to the connection issues in his working station, Randanigala, Dr. Akkalanka will present it in person today. So the paper is by Dr. M.A. Pinidia, W.P.M.K. Abewardana, B.D.S. Jayawardana, G.A.T. Prasad, C.G. Vijayasinghe, R.R.M.K.K. Vijayasundara, and L.G.S. Lokugalappatthi.
Good afternoon, everyone. I'm Dr. Akkalanka from Department of Wildlife Conservation. So I'm going to present about very interesting and very important wildlife issue. It is about mortality, rescue, and release statistic of Panthera pardus scotia in central hills of Sri Lanka during 2019 and 2020. First, I would like to do some introduction about the leopard, Sri Lankan leopard. So, as you all know about, they are under the kingdom Animalia, and the species is called Panthera pardus. We have one subspecies, it is called Panthera pardus scotia. And if you got the cat family, normally we are describing two cat family, big cat family and small cat family. If we go to the world situation, there are 15 big cat families are there, and other 33 of small cat families also there. In Sri Lanka, we have only big cat family. It is the Panthera pardus scotia, and other than that, we have only 3 small cat family. You all know about rusty spotted cat. They are the world's smallest cat, like that. And Panthera pardus scotia subspecies of the leopard is endemic to Sri Lanka, and they are under the listed under the vulnerable category by IUCN since 2020. And if you talk about their distribution, you know they are living various types of climatic zone in Sri Lanka, especially dry zone, Yala, Vilpattu, Kilinoki, like that. And other than that, they are living wet zone, especially in Singaraja and Denia area. And another important thing area is the central hill area of the Sri Lanka. Normally, with this study, we try to give our details about the central hill area. It contains several districts like especially Nuwara district, Kandy, Matale, Badulla, and Kegol district. And this is the recent done something. Actually, it is leopard incidents. It means leopard attack, maybe human attack, maybe or livestock or domestic animal attack. If you go through this, you can clearly understand there most incidents will happen outside of the protected area. It means they are with the human area. In this article, we mainly focusing on leopard death, leopard rescue, and leopard release in central hill area of the Sri Lanka during 2019 and 2020. If you talk about the leopard deaths and casualties, we have encountered within these 2 years totally 24 leopard deaths and casualties. In 2019, we had only 6 cases with 3 male and with 3 female, and 2020 it was drastically increased up to 18 cases with 11 male and 7 female. And those are the leopard deaths in 2019. Nuwara Hatton area. Most of what happened within the estate area, and several cases cause of death were there, and age were 3 years old, 7, 4 years old, like that. And if you talk about cause of death, 2 snare cases were reported. It is the most common problematic case in the snare case with leopard, and 1 gunshot and 1 infection case, but we couldn't identify what is the exact pathogen. We suspect it is viral infection because we submitted only histopathological samples only, and 1 poisoning case. If we got gender variation, 3 females were there and 2 males were there. And then let's move to the leopard deaths in 2020. We had more leopard deaths in last year. In it is same also in the estate area, and most of were snare cases, and age were 1 year, 9, 4, 5, 6, 7 age were there. And look at the cause of death. 10 snare cases were there and 1 gunshot. So other than that, if we talk about gender, 5 male and 5 female. 1 case cannot identify because the carcass was decomposed. And here you can see some photographs with we did in the postmortem. And mostly it's common thing is the snare. Normally snare place up 2 places in the body of those leopards, especially in lumbar area and the thoracic area. Mainly upper 2 photographs, you can clearly see those upper area, lumbar region, the lumbar muscle bleeding, and down one you can see lung bleeding. It means thoracic, based on the thoracic area. And Other one is the gunshot wound. Actually, it's a head damage. You can see bleeding, muscle bleeding, and some area. And this is the very important part, rescue and release of those studies. And we have encountered within those years 24 cases. Out of 24 cases, we were attend only 11 live cases. It means other 13 were already dead cases. From those 11 cases, 8 leopards were successfully released to their natural habitat after the treatment procedures with our intervention. So it means only 11 out of 11 cases, only 3 cases were died within the rescue process or after the rescuing operation. So those are the rescue places and the animal details. All of are in snare cases, and we released 2 female and 6 male, and age were 1.5 year, 6 year, 3.5 year, like that. It is vary. And this is the sum of rescue operation we had done it Ginigat Plain area. Please watch it. And this is the final part of my presentation. And there are several suggestions are there. We should increase public awareness. Actually, most of the things happen within the estates area. So we should educate those people with about their. We should educate them about their behavior, leopard behavior, like that. And second one is we should strengthen laws and regulation as a wildlife crime. So other important thing is improve modern veterinary facilities for if there's any emergency cases, and we should conduct adequate research about especially in the upcountry leopard, because we have limited research data on those animals. And finally, I would like to especially thank you for the wildlife officers who are working with several difficulties for the innocent wild animals. And thank you very much for attending attention. Thank you very much.
Thank you, Dr. Akalanka. Yeah, very nice presentation. So the paper is open for discussion and questions now.
Thank you very much, Dr. Akalanka. It was really interesting to, and thank you for giving us a real wildlife experience. And just for the interest, do you have any idea why there's a drastic increment of cases in 2020 compared to 2019?
Yeah, I have one comment. I don't know if it's correct or not. In 2019, we had only 6 cases. I think it is normal. Normal means we have reported 6-7 cases normal. But you can remember in the COVID situation in 2020 earlier, there was no any COVID incident in the estate area at that time. But most of the time, the female workers are working in the estate, male are working in the construction site or other areas in the outside of the estate, mainly Colombo. And that time, there were only things that were closed. Then they came to the their houses, and they started doing the putting some snares or things. I think this is the situation. I cannot say exactly. This is my comment.
Yeah.
Population count of these leopards in different geographical areas.
Yes, we had done in some researchers had done in Yala actually, especially in the Yala block 1 only. They placed some trail cameras, the trail cameras. But other areas we don't have done any counting. It's very difficult. We should definitely we should use high number of trail cameras and the software to identify the trends.
At least how many entire country do you have some?
They said I think 1,200 maybe like that.
Oh, that's fine, no?
Yeah, but it's not correct. I don't know.
7, 8 days every year.
Yes. Yeah.
I can remember in Porton Plains, where especially in Nagra, Bopat, and they were quite large number of
Yeah. Yeah. Also in the VRR, if you go to the Kandy to in the evening or 8 p.m. from Kandy to Badulla, you can see some 1 or 2 leopards within the main road in the Randanigal area. It's clear, it's common. No. Yes, madam.
Why would they set snares? Just to protect their animals or people, or are there any other?
Actually, it is for meat,
reason?
man. For consumption. They don't mainly target on leopard. They target on wild boar, porcupine, rabbit, so deer like that. But leopards are the suffering.
they don't necessarily target leopards.
I see. I see.
I have seen video footages on television. Yeah, dogs, domestic dogs.
Yes.
It's another line that especially.
Just one question. But about the poisoning case, was it accidental poisoning or was it done purposefully?
I don't have clear idea about that because I got only target, and 1 week later we were there, but we didn't get any idea about that. That is, I think it's sometime the leopards were coming to cattle shed. Sometimes they put some poison.
And were you able to identify the type of poison they had?
Actually, it was a little bit older carcass. That's the problem. Same.
Yeah. Okay. Thank you, Dr. Francis.
Can I ask one question?
Yes, sir.
Thank you very much for that very interesting presentation, Akalanka. So my question is, after releasing this animal, is there a way to observe them for some time, whether they are doing well or because?
Yes, sir. Actually, the main, the ideal thing is we should put a GPS collar or something. But in that time, we don't have those collars or something. But normally, if we release some animal in that place, some place, we physically observe 2-3 days in those areas. And normally we try to put the trail cameras. Actually, we can get idea about the animal movement and the things and the photos.
Because it is not only the
Yes. Yeah.
After the release also.
Yes. Yes.
Thank you.
Thank you, Dr. Akalanka.
Thank you.
So we'll move. Another question? No, I think it's the record. Next question. Arisa? Dr. Indunil?
Thank you. Hope you can hear me well, and thank you for giving me the chance. Just a suggestion, not directly related to this presentation. I mean, being in the kind of outside from from the scene, that just a suggestion to this wildlife team, that because they are doing a kind of marvelous job, and just to have a kind of this kind of work, I suggest to have a kind of small video, and maybe to release to media, because otherwise we always hear that maybe role of wildlife in a kind of disastrous situation, or maybe a kind of with a negative impact on the role of veterinarian. Because they are doing a kind of marvelous job throughout the year, and because of the public and media interest, if you make a kind of small video or this kind of briefing, so then definitely they would publish these. Then public will know that we have as veterinarians, we are doing a kind of great role, other than only handling these kind of difficult cases. Just a suggestion to the team wildlife. Thank you.
Okay, thank you very much, sir.
Thank you, Dr. Indra. Yeah, it's a good suggestion, and I hope wildlife department will look into it. So we'll move on to the next presentation. It's on insights into research on infectious diseases in ornamental fish and cultured food fish in Sri Lanka, trends and gaps. Dr. D. Gunaratna and E. Patirana.
All right. Good afternoon, everyone. When COVID-19 kept us at home, one of my students, supervised by me, reviewed research studies on infectious diseases in commercial aquaculture in Sri Lanka, a long felt need. So here we are with our insights. Just to give you a bit of background about our study, the global aquaculture production increased tremendously and rapidly over the past decades. Although Sri Lanka is not a big player in the global aquaculture trade, we have demonstrated increase in ornamental fish exports throughout the years. So irrespective of the position of a country in this trade, all countries are continuously experiencing challenges with health management. The position of Sri Lanka in this global aquaculture trade is again largely determined by the disease-free status of our fish stocks. So the objective of our study was to review research reporting infectious diseases in cultured ornamental fish and cultured food fish in Sri Lanka. And we selected 24 research publications addressing infectious diseases in Sri Lankan aquaculture, and also publications reporting the presence of infectious agents in fish originating from Sri Lanka. And we used 4 keys to analyze the results of these publications. One was the frequency of different groups of infectious studies that have been researched, and then the species of fish that were commonly affected by these diseases, and the screening and diagnostic approaches that have been taken to study these diseases, and also the efficacy of treatment protocols, particularly antibiotic resistance. So to give you an overview of the results of our study, both bacterial and parasitic diseases were equally researched, while the viral infections were less studied, or they occupied a lesser percentage. And also these studies that were on viral infections were merely presenting, they were surveillance studies that were merely reporting the presence or absence of a virus. And when we looked at the parasitic diseases, there were a wide range of parasites that were encountered in ornamental fish. And there were only 2 studies out of the 24 which reported parasites in food fish. So going back to the parasitic diseases in ornamental fish, all these species of fish had either one or many types of parasites in them. And these parasites ranged, there were protozoan parasites, starting from Tetrahymena all the way down to Pisidium. And then there were monogenian trematodes. Now we all are familiar with Dactylogyrus and Gyrodactylus, but there were much less spoken monogenian trematodes that were reported in fish imported from Sri Lanka. Of course, it was not a local study, but this struck an alarm that we need more comprehensive parasitic studies to investigate the parasites present in our ornamental fish before they are being exported. Then there were digenean trematodes like Centrocystis species and yellow grub, parasitic crustaceans like Arguellus, Ergocillus, and Lernia, then nematodes like Capillaria species. Then moving to the parasitic diseases in food fish, although there were only 2 studies, they reported parasites in a range of food fish species, and they were actually parasitic copypods. Then moving to the bacterial pathogens that were researched or were associated with diseased fish in Sri Lankan aquaculture, Aeromonas, Mycobacterium, and Vibrio species were the most commonly studied. And Aeromonas species were commonly isolated. Actually, there were out of the studies that we took into our study, 25% of them reported Aeromonas, and half of these studies that have isolated Aeromonas employed molecular methods to characterize these isolates. So they have used molecular methods to screen Aeromonas from the preliminary isolates, and the results were quite variable. There were one study reported the dominance of Aeromonas veronii, 79.2%, but then there were other studies reporting that Aeromonas hydrophila were dominating. Then talking about the antibiotic resistance, these same Aeromonas isolates showed resistance to a range of antibiotics, starting from tetracyclines, penicillins, particularly there were studies reporting resistance to ampicillin, then trimethoprim sulfa and erythromycin, which is, now to see these results, it's a bit scary. And the only reported sensitivity was for quinolones. Then talking about the viral diseases, actually the status of viral infections in Sri Lankan aquaculture was quite ambiguous. There was one surveillance study in a fish breeder farm, which concluded that Sri Lanka may be free from spring viremia of carp virus. But then there was another study which indicated the suspected presence. It was stated as suspected presence of SVC, SVCV, and 2 other common fish viruses. Then megalocytic virus was present in gurami exported to Australia, which was detected during border quarantine. So these findings, particularly the last finding, highlights the importance of revising or reconsidering our pre-export veterinary certification procedures. Of course, there's a chance for these fish to be negative during our testing procedures and after an episode of stress because of transportation, they might start secreting virus. but we still need to think of better diagnostic methods to make sure that we are exporting fish stocks which are free from these viruses of concern. To conclude our study, it is required to conduct conclusive surveillance studies on commonly encountered fish viruses. Again, a long felt need, in my view. And also we should have comprehensive viral and parasitic studies, particularly in the ornamental fish export trade, to make sure that we don't miss out most of the viruses and parasites that can be there in our fish exports. Thank you very much for your attention.
Thank you, Dr. Patirana. The paper is open for discussion now.
I have a small comment to our own presentation, because just now I heard an interesting presentation reporting ISKNV in fish, which is interesting and encouraging. I think in future we will have a better idea about the viral infection status of our aquaculture. Thank you.
Thank you, Eran.
Thank you, Erandi, for that.
Thank you,
so
sir.
according to your finding, there is less information on viral diseases, right? So as a virologist, I have a reason for that. So sometimes we find certain diseases, but we are careful enough not to report them, not to publish them. But we inform the relevant authority that disease is there because we don't want to kill the industry. So I want to give one example. This is only for vets here. You know, koi herpes virus. So if you detect it, we cannot export our ornamental fish into any of these countries. So recently we detected it, but as scientists, we did not go and publish it because we know if we did that, that will kill our industry. So what we did was we wrote to the Director General informing that disease is present in this area and please do the needful to control the disease. So what you see in the literature is not the real picture. There are a lot of work going behind the scene, and being responsible scientists, we are not going to publish each and every those cases. This is for your information.
Yes, thank you, sir. I totally agree with you. But the thing is, I was, the laboratory I worked for my PhD was the OI reference laboratory for iridoviruses. So what I saw was sometimes the fish they get from Sri Lanka, like they used to, the border quarantine used to send samples to our laboratory, and then they become positive. So my concern is that even if we keep silent, would it be something good in the long run?
So no, I am not telling we are keeping silent. We are not reporting, but we inform the relevant authority, those who are having the power and legal authority, legal power to deal with that one, instead of jumping and reporting and sending the message throughout the world. So if we can control it without letting it go out of hand, it will be better for the industry. That's what we are doing. So if we don't report, that is surely going to be detected at one point, because we are exporting and the importing countries going to test them and they will detect at one point. So what we did was, after detecting, we just inform the relevant people to please and give the information, all the information we have. Please go and control it before others detecting it.
Exactly, sir. Exactly. So I think what Dr. Samantika and her team was doing, they have started surveillance studies. So if we have that information, maybe we can help our ornamental fish exporters to get rid of, or actually to control these viruses. Thank you.
I would like, can I have some few minutes to comment as I am involving in the industry, industrial trade? Can I comment?
Yes, Dr. Arundhati, go ahead.
Yeah, actually, Doctor, it is Department of Animal Protection Health already has doing some investigations. Actually, we biannually we do test, and we also come across with. This is not the first time Megalocyte was detected. Dr. Kasagala is doing the test, and we do the surveillances, and we do take some active decision and supportive system to the ornamental industry also. And at the same time, where the koi cups is detected, what I heard is all the fishes were dead. And so there is no more, spreading from that farm outside the thing. So we keep on testing whenever we export, and especially when a new ornamental fish farmer is trying to export, we do all the, say, required tests, space, SVC, koi cups and Migelo, depending on the country requirements. We do carry on this is not the first time. It has been carrying out, on a routine basis by the Department of Animal Protection Health. As Dr. Mallavarachi has come out, we don't show the thing out, right? But we try to, do the surveillance. Actually, I'm coming down the real fact. The cost for the testing is quite high. So now anyway, with the with the current reasoning also, DG has meeting to have a more surveillance, more tight, more screwed in the routine testing. But with the COVID and all, it's everything is a bit dispersed. But we do on a track. That's what I can tell you all. Thank you for all the concerns.
Thank you, Dr. Arundhati. Yeah, it's great to learn that things are moving on. So yeah, we would be able to do something to help protect our ornamental export industry. Thank you.
Thank you.
Thank you, Dr. Randi, for your interesting presentation again, and all the participants who actively participate at the discussion and share your thoughts and experiences. So we'll move on to the next presentation. It's on occurrence of Paradivichardia, the Divichardia helicosis in a dead dugong in Kilinochchi, Sri Lanka. It will be presented by PTID Amarasiri, MDS De Silva, B. Giridharan, R. Gaurichilakan, AMH Chatapattu, and NC Gamagedara.
I am Iromi Amarasiri from Parasitology Division of Veterinary Research Institute. This presentation is based on a study conducted on occurrence of Paradivichardia helicoris in a dead dugong in Kilinochchi, Sri Lanka. Dugong, generally known as dugong or Muhuduura in Sinhala, is a marine mammal, and according to IUCN Red List of Threatened Species, dugong is a critically endangered species in Sri Lanka. Therefore, dugong is strictly protected under Fauna and Flora Protection Ordinance. So this marine mammal plays a very important role in maintaining coastal ecosystem. Paradivichardia helicoris of family Ascarididae and genus Paradivichardia is the only nematode present in the stomach of dugong. Although lesser number of parasite doesn't cause much harm, heavy parasitic infection can lead to inflammation and ulceration of gastric wall and also obstruction of proximal part of the duodenum, leading to death of the host. Therefore, knowledge on occurrence of this parasite is very much important in dugongs in order to take appropriate conservation methods. So the objective of this study was morphological identification of Paradivichardia helicoris in dugong present in coastal environment around Sri Lanka. The necropsy was performed at Kilinochchi, Sri Lanka, and during necropsy, some nematode parasites were observed in cardiac part of the stomach as well as in proximal duodenum. And a few of the nematodes were collected as a sample and were fixed in 70% alcohol and sent to the laboratory for identification. So in the laboratory, total of 4 parasites were observed macroscopically and microscopically to identify morphological characters. And the identification was performed using key characters described in previous studies. So the parasites were white in color, macroscopic, averaged 10.3 centimeters. So microscopic features of anterior end include well-demarcated head from rest of the body, and head had 3 clear lips where subventral lips are larger than the dorsal lip. And there was an excretory notch just below the head, and there was an esophagus which was lengthened 1 to 1.5 centimeter. Examination of posterior end revealed the parasites to be non-bursate with very short pair of spicules with no gobeniculum. And anus was really closer to the posterior end with some papillae, and the average length from lips to the anus was around 10.17 centimeters. So this image shows the nematode parasites observed at necropsy, and this is the microscopic appearance observed at the laboratory. And this is the microscopic appearance of anterior end of the parasite. So there are 3 lips in the head region. Subventral lips are larger than the dorsal lip, and there was an excretory notch just below the head. And this is the microscopic appearance of posterior end of the parasite. As I mentioned before, the parasites were non-bursate with no gobeniculum and short spicules. And this is the anus, which is located really closer to the posterior end. So microscopic and macroscopic characters together with size of isolation suggest the parasite to be Paradivjardenia helicoris. However, molecular identification is targeted to be achieved in near future for the confirmation and to get more information. That is all about my presentation. Thank you very much for your attention.
Thank you, Dr. Amalkiri, for your interesting presentation. Now the paper is open for questions. Just for the interest, Dr. Romi, do you think that microscopic identification of the parasite is not sufficient for going for the confirmatory diagnosis in this case? Please excuse me, just want to make an announcement. If any of these authors or co-authors are joining us live online, so can you please raise your hand so we can unmute your microphone? Okay, Dr. Sharika, go ahead. Sorry. Yeah, what I was asking is that just the morphological identification of the parasite is not sufficient for confirmation of this parasite.
Can you hear me, Dr. Sharika? Can you hear me?
Yes, we can hear you. Go ahead.
Actually, there are very little literature available about this parasite.
You are not very clear, Iromi. Okay, Iromi.
Can you hear me now?
Yes, okay.
The available literature is very much limited about these parasites, and the morphology is described in very few papers. So, and I noted few characters described in those papers. I think it is it's good for go for a molecular identification.
I also would like to comment because I did the dugong postmortem. I think maybe and I presented also. And I also have seen visible for these parasites, but when I search for the literature, I found there are some, literature says these parasites are normally they found and it doesn't do much harm. I'm not, counter telling anything, but what I refer at the time, and even they were plenty, and but the both dugongs are very fully healthy, but they were heavily invested with the parasite, but they didn't show any signs of malnutrition or anything. Just a comment on this, not contradict anything. Okay, thank you.
Okay.
Thank you, Dr. Shalika. Do you want to ask anything more?
No, thank you. May I ask a question?
Yes, madam.
Yeah. Now, Dr. Amarasiri, what is the cause of death of this dugong?
Obviously, it is not the parasitic, madam.
Can you repeat it, Dr. Romi?
It is not due to parasitic, madam. It is due to mechanical injury, madam. Madam, may I repeat, madam?
Mechanical reason.
Is it a strike of a trawler or something? Fishing vessel strike or something?
Yes, madam. Yes, maybe due to a boat attack by a boat or something, madam.
In the previous post-mortem that Dr. Arundhati mentioned, we were told that it was caused by explosion of dynamite. The fishermen used dynamite.
Yes, ma'am. Yes, ma'am.
So, yeah, that is one of the causes of death of dugong.
Dr. Romi, but do you find any clinical significance of this Paraduchardenia helicosis in dugongs? Or like Dr. Arundhati mentioned, could it be just incidental finding?
Madam, certain papers the parasitic infestation can lead to death of the host. And since being an endangered species, I think this knowledge is important in species. There's a very limited number of Indian Ocean, I think now.
Okay,
it is described as madam.
Yeah, Dr. Romi?
Yes, madam.
Yeah. You were not very clear. Could you please repeat it?
Most certain papers describe that, madam, it can be a cause of death if the parasitic burden is really high. And since the dugong is an endangered species, so I think this knowledge is very important in conserving dugongs.
Yes. Yeah, it's very important because there are very few reports and very few studies done on dugongs in Sri Lanka. So any finding is important, and it's very important to record it. So thank you so much for your presentation. And then we'll move on to the next presentation. It's on it's a retrospective analysis of ornamental fish submission to university-based fish disease diagnostic laboratory by RM IM Ratnayaka, DM PM Dhanapala, KLN Ananda, AW Kalupahana, and SSSDS Jagoda.
Hello everyone, I am going to present about a retrospective analysis of ornamental fish submissions to a university-based fish disease diagnostic laboratory. Center for Aquatic Animal Disease Diagnosis and Research, OCADA, is a fish disease diagnostic laboratory attached to the Faculty of Veterinary Medicine and Animal Science. One of its major services is providing fish disease diagnostic services to the general public. The objective of this study was to analyze clinical and diagnostic findings of ornamental fish cases submitted to CADA. A retrospective analysis of written laboratory records of 110 cases submitted for disease investigation from July 2017 to July 2021 was conducted. Following data were extracted from the records and analyzed. The source of submitted fish, type of fish species, clinical signs, etiological agent based on the diagnosis. Out of the 110 cases submitted, 3 major sources were identified. 75% of the cases were submitted by hobbyists and 13% were submitted by farms. 12% were submitted by aquaria. Submitted fish belong to 12 species, among which koi carp was the most frequently submitted species, which is at 23% of the total cases. Giant gourami was 14% of the total cases and 13% was goldfish. 8% of guppy, 7% of oscar and 6% of arowana were present. In addition, catfish, fighterfish, swordtail, platy, discus, and flowerhorn were the other species of fish that were presented. This is an image of a koi carp, and this is an image of a giant gourami that was submitted to our lab. This is an image of a goldfish and an arowana that was submitted to our lab, and some images of oscar and guppies. When considering the history, external examination, necropsy findings, and the results of other diagnostic tests, we categorize the submitted cases into 3 major types, among which the majority of the cases were infectious cases, which is a 92% of the total cases. 6% was tumors and 2% were environmental cases. This is an image of a giant gourami infested with saprophyllomania, which is an infectious cause, and this is an image of a giant gourami with toxicity, which is an environmental cause, and this is a goldfish with a tumor. This is a giant gourami with anchorworm infestation, and the red patches all over the body are the places where the anchorworm was attached. This is a goldfish with a fibroma in its caudal peduncle and a catfish with tumor. Majority of the cases were submitted to investigate the sudden and or heavy mortalities, which was at 24% of the total cases submitted. The most prominent clinical signs of the submitted cases were reduced appetite, which was 14%, and 13% of dropsy, 10% of abnormal swimming, 9% lethargy, 7% ulceration, 6% hemorrhages on the skin, and 5% of fin rot. This is an image of a goldfish with dropsy and a giant gourami with abnormal swimming. This is a catfish with ulceration, a giant gourami with fin rot on the edges of its fins, and a goldfish with hemorrhages. When the infectious causes were considered, majority of the infectious cases were due to a single etiology, which was 89% of the cases. 11% were due to multifactorial etiology. When the single etiology cases were further analyzed, more than half of it were due to external parasite, which is 48% of the 89%. And 32% were due to systemic bacteria, 7% due to viruses, and 2% due to external bacteria. When the multifactorial etiology infectious cases were analyzed, 5% of it was parasites and systemic bacteria, 3% was systemic bacteria and viruses, 2% was parasites and viruses, another 2% was external bacteria and fungi, and 1% was parasites and fungi. When the parasitic infestations were analyzed, 50% of the submitted cases had parasitic infestations, and the majority of the cases were due to monogenians, which was a 47% of the total parasitic infestation cases. 20% was due to trichodina, 9% Lernia, 9% Udinium, 7% Ichthyopteris, 7% Metacercariae, 5% Ichthyopoda, 5% Epistylis, and 4% Chilodinella. This is an image of monogenians from a wet mount of a skin scraping. They are commonly known as flukes. This is an image of Trichodina. This is Udinium, and this is Ichthyopteris, which is also known as white spot. Among the 110 cases, 36% was systemic infections, and out of these systemic infections, 56% was due to gram-negative bacteria. 28% were due to mycobacteria, and 16% were due to gram-positive bacteria. This is an image of gram-negative rods, and this is an image of mycobacteria stained with Ziehl-Neelsen stain. The acid-fast positive mycobacteria are displayed in pink color. 11 out of 110 cases were due to viral diseases, and we confirmed these causes by doing PCR using specific primers. 42% was due to Carpedema virus, 33% due to red sea bream iridovirus, and 25% due to megalocytovirus. This is an image of the fish affected by Carpedema virus, and the disease is called koi sleeping disease. The results show the frequent occurrence of external parasites and bacteria associated with diseased freshwater ornamental fish. Among the samples submitted, only 13% were from farms. This could be due to increased distance that act as a barrier to access to the laboratory because of increased travel time. In conclusion, with the expansion of ornamental fish industry, there will be an increasing demand on quality diagnostic support. A successful outcome is possible when logical treatment strategies are initiated and supported by diagnostic testing. A concentrated effort in improving diagnostic support will be immensely beneficial to the ornamental fish industry. I would like to acknowledge all the academic and non-academic staff members of CADA. Thank you all for your attention.
Thank you, Dr. Ratnayaka. Now the paper is open for questions. Authors who are joining with us online, please raise your hand so we can unmute your microphone. What were the types of tumors that you have encountered in this fish?
I'm sorry, madam.
What were the types of tumors that has been diagnosed in these fish?
We really didn't go for the diagnosis, madam. We observed the impression smears of some tumors, but didn't identify a specific type of tumor, madam. And in the catfish, it was diagnosed as a melanoma, after the necropsy, madam.
Okay. And also you have said some of the causes were due to environmental causes. So,
Yes.
So what were these causes?
We suspected 2 cases of toxicity. The owners has suspected adding some toxins to the water, and all the fish in the pond has died.
Okay, only toxicity or any other causes as well? Like in other cases?
No, madam.
Okay, thank you.
Thank you, madam.
Akhilesh,
Yes,
these
madam.
fish, where have they come from or brought from? Around Kandy or
Mostly around candy from hobbies, madam. Some call and contact us and come from around various parts of the country, and the farms we get the samples from NACDA and the other ornamental farms in the all around the country. Hobbies mainly around candy, madam.
So do you recommend treatment or any preventive measures as well?
Yes, madam. Treatment to the hobbyists. We hospitalize the fish when we can, or if they can take the fish at home, to the home, we recommend treatment, madam.
Okay, thank you.
Thank you, madam.
Thank you. Then we'll move on to the next paper. It's on evaluation of nutritional composition of sucker-mouth armored catfish, Serigolipsis disjunctivus, collected from reservoirs in Sri Lanka by WMLA Vanasinghe, MNM Fauzi, LGS Lokugalapatti, HDS Aryaratna, and MWCD Palyaguru.
Good morning to everyone. It's happy to meet you to discuss about one of our project on nutritional analysis of sucker mouth armored catfish collected from the different reservoirs in Sri Lanka. We have collected sample from many reservoirs. These fish we call they are from the Loricaridae and the genus Pterigopistis. And this fish is commonly known as sucker mouth armored catfish, although they have they are called by different names: tank cleaners, scavenger fish, sailfin catfish, vermiculated catfish, South American sailfin catfish. They are highly invasive and accidentally introduced to freshwater bodies. So when you say invasive fish is an organism that was ecological and economical harmful to the new environment where it is not native. So this fish is established as an invasive fish in tropical, subtropical system around the world. For example, southern United States, Caribbean, Mexico, South Africa, Philippines, Taiwan, and other parts of South Asia, including Sri Lanka. So it is a serious matter, serious effect it was, and it was adverse effects of invasive fish, invasive Pterigopistis disjunctivus. That it causes change in the habitat structure and establish the river banks and depletion of native freshwater species diversity and ability to tolerate. So this is because they are ability to tolerate the polluted environments. And so they, especially it causes the damages the gillnet of the fishermen. So therefore, it causes big effect on the local fisheries, and it is a lack of marketing value as a food fish. Although the fishermen catch mostly this kind of a fish in our reservoirs, but they cannot market them because very low consumer demand. So therefore, this fish has to be controlled. This invasive fish has to be controlled. One of our suggestions is to use them as a other edible source of for human consumption. It is very hardly you can convince the people to consume. Therefore, it can be used as an animal feed. So with this hypothesis, we had the objective to determine the nutritional composition, including food fiber, protein, ash, fat, mineral content, and fatty acid profile of Pterygopolis distinctivus in freshwater bodies of Sri Lanka. So for this, we have collected the sample from many reservoirs. Especially, we collected sample from pollular reservoirs, which were mainly used for proximate analysis. In addition to the pollular reservoir, we also collected sample from Victoria, Udavalava, Halava, and Janaranjana reservoir from Tringamalay. So these fish were used for mineral analysis. So all the fish which were identified, which were caught, were identified or they were classified according to the taxonomic key, which were prepared by the already established key, and we have categorized them, and we have used only this particular species. There were other confusing species, so we all first confirm this species, then only we use this species for nutritional analysis. So the samples, all fish were anesthetized and in immersing fish in ice for 4 hours, and they were euthanized. So the euthanized methods were already proposed to the ethical committee, the faculty, and it was approved, and we got the ethical clearance certification. Then they were, according to this method, we euthanized them, and we collected the sample, especially the muscle tissue only we use for the nutritional analysis. Head and the viscera were discarded. So crude fiber, protein, ash, mineral, fat, and fatty acids profiles were analyzed by using these samples. Crude fiber content were analyzed by digesting the sample in sulfuric acids and drops of octanol, and it was undergone for these different steps, and finally we were able to come to the to quantify the amount or the content of the crude fiber. Similarly, the protein content also was measured by using determined by using the Kjeldahl method. This Kjeldahl method was very one of the very popular method to analyze the protein content. Ash content was determined by weighing the sample and digest the sample or the process sample in a muffled furnace at 550 degrees centigrade for 6 hours. So then the ash were got and we analyzed. Fat content fat was extracted into ether and methylated to fatty acid methyl esters, FAM, using methanol. This FAM was quantitatively measured by capillary gas chromatography against C11 or 0 or internal standard. So by this method, the total fat was calculated. Fatty acid profile analysis was determined by using AOAC 99606 method. By this method, we were able to calculate, able to measure saturated and monosaturated fats. And mineral contents was also analyzed by using the atomic absorption spectrometer method in VRI. So the data were obtained and analyzed by one-way ANOVA test by using SPSS software, and we use p-value less than 0.05 as a significant value. So we got the results, impressive results, that the average moisture content of the fish was 73%. However, as the fish is ultimately used for fish meal, which is a dry product, the rest of the nutrients are on dry matter basis. So on dry matter basis, we got the protein content was 61 or over 60%, and fat was also 14%. Crude fiber also we got the less than 10%. So we have analyzed. We have our statistical analysis showed there was no significant difference between the weight groups in the nutritional composition. Whether the ash, protein, fat, food fiber, within the weight group, there was no significant difference. So all these contents were same in all different weight groups. And the mineral content was, significant mineral content was found. Calcium, phosphorus, sodium, potassium, manganese, zinc. We got these all, different reservoirs. But mainly Victoria reservoir in the calcium and phosphorus was high, whereas manganese and zinc was high in other Polgola reservoir. And Kalawava reservoir, we found least amount of these minerals. So fatty acid analysis, explained and resulted with the palmitic acid as a saturated fat was significantly high. And similarly, oleic acid as a monounsaturated fat also was high. And then linoleic acid also we found a significant amount, 6.5% as a polyunsaturated fat. So this is we have analyzed this fatty acid. There was no. So let us see the discussion where we have found the crude fiber content is 4.09. Okay, in the present study from this species, which is much more higher than the fish like sapu fish. Okay, sapu fish we have when you compare with the already published data, a little higher. And because we have got in the tissue there may be more fiber, so the fiber content is higher here. But importantly, the protein in the muscles, although with the muscle part in the fish is comparatively low, but the protein is very high in this muscle tissue. It is 60.9. It's almost 61%. Okay, on dry matter basis. So which is very high among our normal freshwater fish, which we are we are using. Okay, so this is a very important fact that we got it. And furthermore, the ash content is also a little higher than other carps and rainbow trout and tilapia. And the fat contents in the suckermouth armored catfish was, among the all fatty fish, the fat content was less. Okay, fat content was less. So let's see now in the sapu, Asian red-tailed catfish, and pomfret, catfish, okay, tilapia, common carp, all these, among these all fatty fish, the suckermouth armored catfish has low content of fat fish, which is similar to that of a raw. And the mineral contents also, when we compare with other publications, we have these three species we compared, where the mineral content, calcium and phosphorus, is much more higher in the suckermouth armored catfish. Okay, and sodium and potassium also bit higher. So this is so as a conclusion of our study, This study shows that the investigated fish species are good source of nutrients, especially protein, minerals, such as calcium, phosphorus, sodium, and potassium. Therefore, this armored catfish, this sailfin armored catfish, can be used as a raw material for protein and minerals in animal feed. So, thank you very much. We are acknowledged to our, 3 projects. We got the funds from the 3 projects, and these are our key references. Thank you very much for listening.
Thank you, Dr. Fauzi. Now the paper is open for any question. Go ahead.
Thank you very much, Dr. Fauzi. I think it's very interesting. I think these tank cleaners have become a nuisance in blocking all the reservoirs as well. If you can use that animal.
Excuse me, the sound is not clear. I cannot hear.
Fauzi, can you hear us?
Yeah.
Okay.
Right. For your nutritional analysis, did you use mussels or entire fish? I know for proteins you use mussels. What about for the other parameters? Did you use mussel or entire fish?
So we use only mussels, because if we use entire fish, definitely the calcium and phosphorus will be high, because this fish has high, the bony fish. So we use only the mussels.
All right. Yeah. That may be why your protein content is so high.
Yeah.
And I wonder why ash content is so high as well. And then number 2 is for your comparison. I'm sure you have done ANOVA, but in your graphs, what did you use as error bars?
Yeah, we use for error bars, we use standard error.
Standard error of mean.
Yeah.
Yeah, because when you say, there are significant differences, but I saw, the error bars all over the place. So that's why I was wondering if they use something else as error bars.
Yeah, not the standard deviation, we use the standard error.
Yeah. What do you reckon? This material can be best used in which species of livestock?
So this can be used for, especially for the poultry fish meal, because especially for calcium and phosphorus, they have high, especially for layers. Layers, it would be better source, for animal protein.
Yeah. Then probably for your next study, what you can do is, the aiming that you are looking for poultry as a species, you can analyze the ME value, you can estimate ME value,
Yeah.
and also peptide digestibility of your protein,
Yeah.
and also, if possible, amino acid profile.
Yeah, that's a very good idea, sir. Thank you very much, because we did not think about it, but it is it's a good to forward our study. Thank you.
Thank you, Dr. Fauzi, for your interesting presentation and the discussion. So we'll move on to the last presentation of today's session. It's on evaluation of export performance of ornamental fish industry in Sri Lanka by US Gunaratna, CS Gangani, and LM Abhivikrama.
Good afternoon, everybody. Thank you for giving this opportunity. My topic is evaluation of export performance of ornamental fish industry in Sri Lanka. Ornamental fish production globally multi-million-dollar industry, and approximately 125 countries involved in this trade. Ornamental fish can be defined attractive, colorful fish of peaceful nature that has kept as pets in confined species of aquarium or garden pool with the purpose of the enjoy their beauty of life. Ornamental fish business has been recognized as a venue employment for women in various countries. Most of the ornamental fish exporters are from developing countries. This is then provide employment opportunity into developing countries. The global ornamental fish market is value $500 billion. Sri Lanka has long history for the ornamental fish. In 1933, there were several small-scale importers and hobbyists. 1952, Export Development Board started the commercial aquarium. According to the facts, 2017, Sri Lanka is the 6th ornamental fish supplier. Ornamental fish species export from Sri Lanka go up to plate and like this. Ornamental fish is higher potential income generating activity in Sri Lanka. Pereira investigated the problem and concern in the ornamental fish. He analytically described the selling price of the ornamental fish is not changing with the changes of the market. Vijay and Aquaculture studied the present status of the future trends of the ornamental fish industry in Sri Lanka. However, studies have conducted on the export performance, the ornamental fish sector of Sri Lanka. Therefore, evaluation of export-oriented fish industry is timely and important. Further studying the export market of the ornamental fish sector of Sri Lanka will be defined, benefit identified marketing opportunities. This study will help to policymakers and stakeholders to develop an emerging study. Objective of this current export performance, export potentials, factor inhibiting exportation. Specific objectives to examine the characteristic of the ornamental fish export sector, understand the export performance, to study the socio-economic background, and to identify the problems and constraints. Limitation of the study. The limitation of the study was finding primary data gathering regarding the income and lack of data in other countries. Research method, I apply mixed method and the data collection, which comprise the literature review, secondary data, and mixed method refers to an emergent methodology research that advances the systemic integration of mixing of quantitative and qualitative data with the single investigation or sustained program inquiry. Primary data collection by the randomly selected fish exporters and 2 exporters were interviewed. And secondary data collection from the Department of Customs, Export Development Board, Census, Statistics, ITC, Uncompleted, and FAO, and in addition to all printed and electronic published documents and the data gathered from the quarantine unit of DAPH. I analyzed according to the RCA, RIA, and RPA. I reveal comparative advantage. Relative import advantage, relative import advantage measures whether focus country has any sort of import advantage in particular product or not. Normally, in the ornamental fish export industry, most of the countries around the world import ornamental fish from the countries and they quarantine animals and export to other countries for higher profits. Therefore, to measure performance, it's important to measure the RIA. Relative advantage means consider both import as well as export for the analysis. The countries with positive RA reveal that the country is highly export-oriented from all resources, and countries with negative RA reveals that they import ornamental fishes from the other nation and they re-export it. Country with positive RA denotes a strong competitor by the own RTA. Present as follows: ornamental fish production of the world. Overall value of the ornamental fish can contribute to world trade is small compared to other sectors. However, the sector plays an important role in terms of poverty alleviation in developing countries and marine preservation. The value of the ornamental fish trade has grown significantly over the past decades. Between 2000 and 2011. Global export of ornamental fish increased like this. Most of the market supply originated from Asia, which Singapore dominates as a top export country. Major exporting countries are Germany, Singapore, China, Hong Kong, like this. Top suppliers of the ornamental fish. Asia contributes the highest export share in other regions such as Europe, South America, and North America, Africa. Supply chain, Singapore is the main country, Japan, Thailand, and Indonesia and Malaysia. These recently they include the Czech Republic, Israel, Belgium, Netherlands. According to the rank of the beauty, the fish can rank like this. Government involvement like the DAPH with the quarantine, wildlife, NACDA and EDB. License need to require for export, health certificate, certificate of the origin, GSP, permit, water quality certificate. Procedure for exportation of ornamental fish, the international trade and animals demand to carry between trading partners in order to avoid the transfer of disease agents that are pre-pathogenic, and animals and humans need to undergo some pre and post-export quarantine from DAPH, and export country should issue an international veterinary health certificate describing the animal health criteria, quantity and farm. In Sri Lanka, according to the Animal Disease Act, the Director General is the responsible for, and it is dedicated to keep animal quarantine. So, when the fly fish are when export checked by the Katanayak airport, checked by the Katanayak quarantine staff, separate checked by the staff from Colombo, animal quarantine officers. Requirement for the health certificate for exporting IVH, BRC, and DAPH monitoring certificates. It's, authorized officers from DAPH should be allowed to visit the farm. Exporter's responsibility. If exporter plan to export restricted fish varieties of permit should be taken by wildlife and fisheries. Wastewater treatment should be done in the prescribed manner. Pre-export quarantine of fish after completion of all requirements about IVH is issued by the quarantine unit, and this valid for 7 days. Results and discussion illustrate the socio-economic characteristic, value of ornamental fish, the contribution of the total export, top export destination, export performance, revealed competitive advantage, revealed import advantage, and trade advantage. According to the interviews conducted with exporter, most of the exporters said that they are small-scale exporters. ADV classification, Sri Lanka, all the exporters was classified as small and medium-scale exporters. It is noted that 70% of the interview exporters are in degree holders. The family size 4 to 6. It was noted 55% exporters have more than 10 years. Firstly, they became breeders, and then after that became exporters. However, export directly joined the ornamental fish exporters. Like this export value of Sri Lanka, that means value of the dollars, and percentage of total export of Sri Lanka, and percentage, this figure describes the percentage contribution of ornamental fish sector total export of country from 2010 to 2019. According to the figure, it is clear that 2015 is increased, and 2015 it's a 0.6 percentage. Export destination mainly USA. According to the Export Development Board, United Kingdom, China, and Germany are the competitive importers to our country. Top export destination from our country is mainly German. The figure developed calculation total export value, according to the country, according to the world, the main total export value is respect to countries Singapore and Japan. Export performance, RCA, and RCA is 43.7 in 2018. In ornamental fish export import advantage, so like 8.9, like they check the industry viability and how the industry has maintained its import to measure the relative trade advantage. This reveal its or results of by importing animal fish to the country and we export to the other destination. Using all resources with the export will be benefited to the industry. According to the calculation RT value 2018, this industry can be developed using our own resources within the country. Further, as indicated, the figure 2015 Sri Lanka shows the highest RT value. It's argued that Sri Lanka can ornament fish has a relative export advantage higher than the relative import advantage. So Sri Lanka shows higher potential to develop and complete this industry as a competitive industry in the export market. Problems and countries, traceability, sustainable management along the supply chain, disease, transport facility, destructive fishing method, introduction of exotic fish. Conclusion, in Sri Lanka, most of the fish exporters are small scale, and 70% of the exporters are having degree level. 50% of exporters have more than 10 years experience. According to the department, Sri Lanka earns $6.1 million US. And the export sector contributes 0.13% to the total export from Sri Lanka. And according to EGB, uncomplete Singapore is the main country. In this study, The export performance of normal fish industry Sri Lanka was measured using relative comparative advantage, relative import advantage, and relative trade advantage. It was revealing that during 2010 to 2018, RC value were increased over the years, and 2015, the RC value of the ornamental fish industry was stocked up 42 to 1, down to 29 to 1. According to various RC's particular industry shows RC value more than 1, it's a break-even point, and there's a relative competitive advantage of engaging the industry. Thank you.
Thank you, Dr. Ajisha. The paper is open for the questions now. Can I? Yes, ma'am. Go ahead.
Dr. Gunaratne, it's a very informative presentation.
Thank you, madam.
And you went like a bullet train.
Thank
You
you.
had about 55 slides. It's okay. Congratulations. What I wanted to suggest is to write this, you, as an article to the Sri Lanka Veterinary Journal, because there's a lot of information and it's very important. Would have done a lot of reading, and a lot of work is involved. So I suggest that you write to the Sri Lanka Veterinary Journal.
Okay, thank you, madam.
Thank you.
Appreciate your comment.
Yeah, I think very useful information. Many of our members have no access. I think if we can compile this, that would be very useful.
Okay, if there are no more questions, this marks the end of this session today. So it was there were 12 papers, and 9 were on aquaculture, and out of that 6 was on ornamental fish, and 3 on 2 on food fish, and 1 on mud crabs for mud crab investigation. And there were 2 papers on free living wild animals and 1 paper on captive wild animals. So it was so encouraging to see this number of this much of papers had been submitted this year. So, and as a fast growing industry in Sri Lanka, aquaculture needs more veterinary involvement, and it's good to see the veterinarians are involved in the sector and doing research on the issues related to aquaculture and ornamental fish trade. And the other thing is in wildlife becoming Sri Lanka is a has a very high biodiversity in wild animals, so veterinary involvement is needed there, not only to mitigate health risks to humans and livestock, but also for the conservation of wild animals in Sri Lanka. So I would like to thank the presenters for sharing their valuable experiences and their thoughts with all the veterinarians in the country, and the audience for active participation, the virtual audience as well as the audience in this room. And especially thanks goes to the judges and Dr. Yushika for comparing this session, and all the assistants who technically supported us, and the hotel staff who assisted us to successfully complete this session. So I'm going to wrap up the session and hand over to Dr. Yushika for closing remarks. Thank you.
Thank you, madam. So I think it was a very enthusiastic session today and a very interactive one. We had 12 interesting presentations, and it marks the end of the wildlife and aquaculture session today. And thank you all, the virtual participants as well as the participants today, for participating in this session. Thank you so much. And the clinical session is still in progression, and you can log into the rest of the clinical presentation as well. So thank you so much for participations.